is our sixth floor. We have also laboratories here, and you can spend your first and second year.
So, sorry, the teaching lab, and we have other labs, we also have a darkroom there.
This is our sixth floor. We have also laboratories here, and you can spend your first and second year. This is our sixth floor. We have also laboratories here, and you can spend your first and second year.
We also have pilot plant. That's where food technology.
Laboratories here, and you can spend your first and second year.
Study the science behind food.
After your food is made, we test it in a sensory lab to take some sensory tests.
Yeah, so you can get free food here while also studying science. Yeah, so you can get free food here while also studying science. Yeah, so you can get free food here while also studying science.
We have auditorium and spaces here.
The space is also used when Student Council makes a lot of events. So no worries, because ITRL has a lot of free spaces.
Let's go. This is our auditorium where we held our big classes, conferences, and annual events.
So at the end of every year, usually the Student Council makes an annual event where students can participate as well as perform. Check it out.
That's it for our campus tour.
We hope you know ITRL better now.
See you in ITRL.
Students can participate as well as perform. Check it out.
That's it for our campus tour.
We hope you know ITRL better now.
See you in ITRL.
Students can participate as well as perform. Check it out.
That's it for our campus tour.
We hope you know ITRL better now.
See you in ITRL.
Students can participate as well as perform. Check it out.
That's it for our campus tour.
We hope you know ITRL better now.
See you in ITRL.
Students can participate as well as perform. Check it out.
That's it for our campus tour.
We hope you know ITRL better now.
See you in ITRL.
Students can participate as well as perform. Check it out. Hello? 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 Hello?
followed by a question and answer session. And the next session will be followed by a talk by Mr. John Busnick, the principal consultant from Insight Pharma Consulting LLC, followed by a Q&A session. And the last session will be delivered by Mr. Surya Septefanus. Question and answer. You complete the evaluation survey before you are able to retrieve your e-certificate. Kepada partisipan dan juga peserta, diharapkan untuk tidak mengaktifasi audio dari Zoom ataupun komputer Anda selama webinar berlangsung. Apabila Anda ingin menanyakan pertanyaan, mohon disampaikan melalui Q&A feature di Zoom melalui chat box, atau apabila Anda mengikuti webinar ini melalui YouTube streaming, Anda dapat mengirimkan pertanyaan Anda melalui link cast form PDA yang telah di-share. Selanjutnya, pada saat webinar ini berakhir, kami akan mendistribusikan evaluation survey. Anda diharapkan untuk mengisi survei evaluasi ini sebelum Anda dapat menerima e-sertifikat Anda. Jadi jangan lupa untuk mengisi, sekali lagi, evaluation survey sebelum Anda dapat menerima e-certificate. Thank you for your attention. Now we would officially begin this PDA e-Townhall event, and I would like to invite our head of department, Ms. Raden Leonie Yulita Hartiadi, PhD, as the head of Pharmacy Study Program of Indonesia International Institute for Life Sciences to give her opening speech. Ms. Leonie, the time is.
Okay, thank you, Ms. Petra, for the introduction. Assalamualaikum warahmatullahi wabarakatuh. Selamat pagi, Bapak dan Ibu semua. Pertama-tama saya ingin mengucapkan selamat datang di acara webinar PDA e-Townhall with Richard Johnson, bringing PDA expertise to pharma industry in Indonesia. Saya harap Bapak dan Ibu semua tidak keberatan kalau saya berbicara dalam bahasa Inggris karena perwakilan PDA baik dari PDA Asia Pacific dan PDA Headquarters sudah hadir bersama kita. I3L is a premium life science higher education and research institute that has a vision to be a leading and globally connected interdisciplinary institution that impacts society through science and innovation. It has become one of our mission to contribute to the development of industry in Indonesia by collaborating with industry and also provide trainings and workshops to support the continuous need of education among professionals. On behalf of I3L, I would like to thank the speakers. First, we have Richard Johnson, PDA President and CEO. We also have John Shebusnik, founder and principal of Insight Pharma Consulting LLC. Last but not least, we also have Surya Septefanus, site head of Global Oncolab Pharma. It's a great honor to have all the speakers with us today. I had the opportunity to get to know PDA better through one of our collaborators, Andianto Sutander, who works in a consultant company in Singapore. From there, we invited Dinesh Kokal, who were the president of PDA Singapore chapter at that time, to give a talk in Jakarta in early 2019. And the following month, I attended PDA event where I met Richard Johnson for the first time. And he gave explanation about what PDA does, what PDA concerned about, and what are their strengths. As PDA is an international organization that provides science, technology, and regulatory information for the pharma industry, then it is without a doubt that a PDA could bring pharma industry in Indonesia, especially those who manufacture sterile products, to the next level. And the idea of Richard Johnson giving a talk in Jakarta actually started in early 2020. We even had the flyer ready to be disseminated. However, due to pandemic, we had to cancel the talk. And now I'm really glad that we are finally able to hold this webinar in collaboration with PDA. We hope that the webinar will become a platform where Indonesian professionals in the pharma industry have this chance to get to know PDA better, with an end goal to bring PDA's expertise to Indonesia by establishing a PDA chapter in Indonesia. As an education institution, it is not possible for us to solely establish PDA chapter by ourselves. We would also need support from professional in pharma industry. Therefore, I would like to invite participants attending the webinar today to also take an active role in establishing a PDA chapter in Indonesia. To close up, I do hope that today's webinar, more and more professional will join PDA, and then we can actually have our very own PDA chapter here in Indonesia. Thank you.
Thank you very much, Ms. Leonie, for the opening speech to officially open our event, the PDA e-Townhall event. Now, I would like to invite our first speaker of the day, but before we begin the talk, I would give a brief introduction to our first speaker, Mr. Richard M. Johnson, the President and CEO of the Parenteral Drug Association. Today, he will give a talk with the topic, Future Challenges and Objectives for PDA in Asia Pacific. Mr. Richard Jim Johnson graduated from Marshall University, majoring in biological science for both his master and bachelor degree. He is currently the president and CEO of the Parenteral Drug Association and was the vice president of Global Quality Assurance for Dutch Animal Health Wyatt. He also was the director of Corporate Quality Center of Excellence for Abbott Laboratories. His affiliation includes the Scientific Advisory Board of PDA, the Regulatory Affairs Quality Control Committee, the chairperson in Sterile Aseptic Processing Interest Group, the co-chair of the US Sub-TAG to ISO TC 198, and the member of Pharma Barcode Committee. Mr. Richard M. Johnson, I would like to invite you to begin your talk. The time is yours. Thank you very much.
Thank you for that introduction. I hope you can see my screen.
Yes, sir, we can see your screen. Thank you very much.
Well, thank you again. It is my pleasure to be here. As was indicated, I had looked forward to the chance to come to Indonesia again, but I am glad to be joining with you virtually. I'm going to speak about PDA, what are our activities, and specifically some of our activities and goals in Asia Pacific. But then I will also spend a few moments talking about some current issues in pharmaceutical manufacturing, especially as they relate to the global pandemic, the current global situation. PDA is a not-for-profit organization. We are celebrating our 75th year this year, so it's our diamond anniversary. And as you heard earlier, our mission is to develop scientifically sound, practical, technical information and expertise for advancing pharmaceutical and biopharmaceutical manufacturing science and regulation. And of course, all of this is done so that you, our members, can better serve patients with better products. This has been a very unprecedented period for all of us. Certainly, as I look back on 2020 and even today, this has been a period of global disruption. But I think it's important for us to take a moment to remember all of those who've been directly impacted by this pandemic and by the economic effects. And I also think we should remember to thank those people, our first responders, our healthcare workers, and of course, our pharmaceutical community that has been throughout this period continuing to provide critical medicines, continuity of safe and effective supplies, and ultimately our hope for a future without COVID through the development of vaccines. PDA is a global organization. We do have chapters around the world. And as you can see, we have a number of chapters in Asia Pacific. But as you also heard, we do not yet have a chapter in Indonesia. And I hope that you will learn more and think about getting involved. The way that PDA does things is through a model that we call advocacy and outreach. So, of course, one of our key areas is by sharing knowledge. We do that through a number of ways, including having world-leading conferences. In fact, as we speak, this week, we are having our annual meeting, and we, for the first time, are having it virtually with sessions at different time zones to accommodate a global audience. We also provide a well-known training for people in industry around the world and for regulators. We produce a number of publications that I will talk about more later. And of course, we have chapters and interest groups and other platforms for exchange of information between our members and the public at large. In addition, of course, we try to drive scientific consensus. There are a lot of regulations in the world. There are a lot of rules that we have to follow. Most of those are pretty similar, but the challenge is that there is a lot of room for interpretation. And in science, not all people interpret everything the same way. So we try to achieve consensus. We do that through the development of technical reports. We also do research to try to identify the best practices. And we are now a standards developing organization. We do regularly include regulators in the participating in these activities. All of our technical reports and standards have regulators who participate or act as peer reviewers. In addition, at PDA, we also collaborate with other organizations. So we collaborate with other nonprofit organizations and other groups such as perhaps I3L. And then we believe another key point of what we do is our engagement with regulators around the world. We do joint conferences with regulators, including, I think we're in our 27th year this year of having a joint conference with the US FDA. We've done similar meetings with EMA, with PICS, with ICH. We comment on regulatory documents around the world. And of course, we have to remember, we have regulators who are members. Over 700 regulators are members of PDA. They're participants, they're reviewers of our technical document. And when you become a part of PDA, you have the opportunity to engage with these regulators as well. In 2020, we introduced our newest strategic plan. And I'm going to share with you a few things that were in that strategic plan that will govern our activities from 2020 to 2026. Of course, with the pandemic, we've had to make some adjustments, but these things that I'm going to share with you have not changed. So one of our core principles is that we want to continue to enhance the value of PDA membership by growing and improving the organization globally. We believe that our membership diversity is a strength. And so we want to continue to expand to have members around the world, because when they join with us, they contribute to better information and better knowledge for all of us. We are working to define some new membership and PDA entry pathways to make it more easy for people in other parts of the world to also participate. And we are very focused on strengthening the diversity of our membership and our volunteers, with a special focus on young professionals. You know, I'm not a young professional, but clearly we need the next generation of people to be learning from those of us who've been in the industry for a while, so that in the future we can continue to contribute to society. So we are working on a number of things to try to improve that. We also want to continue to be a premier global leader for agile advancement of science, manufacturing, quality, and innovation. We are very strong in certain areas, and we are expanding into other areas. And as I said, we do that through the development of PDA standards, technical documents, trainings, workshops, and scientific research. We are using on-site and classroom training, and increasingly we are using e-learning for both conferences and for training, especially as a result of the pandemic. We are not only working on our traditional areas, but we're very focused on some of the emerging topics, such as personalized medicine, including regenerative medicine, cell and gene therapy, and combination products, and computer science, digitalization, automation, and artificial intelligence, especially as they apply to pharmaceutical manufacturing. We also have a desire to provide some training on basic and advanced GMP and GDP topics, so that other areas in the world that need this kind of training will have access to it, and to make sure that we're providing these tools and resources to young professionals. Specifically, we want to engage in countries such as Indonesia with growing manufacturing capabilities and educate them on standards and best practices, and also invite them to join our community so that we can learn from them as well. With regard to the pandemic, you know, we certainly had to think about some specific things. So I want to mention a few of those. Shortly after the global pandemic was announced in March of last year, PDA formed a COVID task force, and immediately it began working on developing guidance on manufacturing, supply chain, and control issues related to the pandemic. There have been a number of documents that we have published and we've made available. We have done a number of webinars on these topics, and we are continuing to do so. We have a manufacturing science and operations program that is focused on not just how we assure that we meet regulatory requirements, but how can we truly improve manufacturing science and operation, because that is really the secret to improving the ability to provide these new products to patients around the world. Another growing area is combination products. PDA has been very active in this area for many years, but we have formed a combination product steering committee so that we can try to identify additional areas that we might need to focus on so that we can continue to advance this area. Things like pre-filled syringes, auto injectors, combination diagnostics and drug delivery products, and so on. And finally, but certainly not least, one of the first impacts of the global pandemic was to interfere with the ability to have typical supplier audits or regulatory inspections. And so we rapidly convened a team who started working on best practices for both remote audits for suppliers and for handling regulatory inspections. And in fact, in about 1 week, we are going to be doing a workshop sharing with you some of the things that are that group has identified so far. So please go to our website if you are interested in that and check it out. Specifically, our strategic plan has identified that Asia Pacific is an area that we want to grow. As of today, 18% of our membership is in Asia Pacific. It is certainly a significant region for pharmaceutical manufacturing and is certainly growing in its importance in biological manufacturing. So we want to expand into Asia Pacific, but we don't just want to have people join PDA. We want to have them participate in PDA so that they can become partners in our efforts to expand our body of knowledge. So I mentioned global conferences. Today, we are in the midst of our annual meeting. In fact, there's a Asia Pacific time zone session that is ongoing in about about 3 minutes. So hopefully if you missed that, you can still join. All of the presentations are available pre-recorded, and there will be some live Q&A that you can access. But as I mentioned, we have a remote audit and inspection workshop coming up that will be made available again online, so it's accessible worldwide. Later in April, we have our visual inspection forum. John Shabushnik, who will be speaking a little bit later in your program, has been the leader of that effort within PDA for many years. And a little bit later in April, we will have our parenteral packaging conference, which again is being offered virtually and will be available to participants around the world. All of these sessions will include segments that are timed to be compatible with the Asia Pacific time zone. And all of them will have conference presentations that are available on demand, either before, during, or after the conference. We have established an Asia Pacific headquarters in Singapore, and our Asia Pacific team is organizing a rebroadcast of sessions from global conferences with some local participants who will be providing some additional local interpretation. In April, they will have a week of Parenteral Learning Week. That will be 4 days. It's all virtual, and it will include both a webinar and some training. In May, there will be a similar learning week focused on sterile manufacturing. And in June, there will be a week focused on pharmaceutical packaging. So please look for those. And if you don't get a chance to participate in the global event, this is an excellent opportunity to gather some of the key learnings from those events. In addition, we will be having some live events in Asia Pacific in the later part of the year. In September in Singapore, we will be having what we've done annually. We will be having a conference. We hope that it will be able to be live, but it will also be offered virtually. So it's what we call a hybrid conference. That will be a 2-day conference in September. In October, there will be a 2-day conference in South Korea on aseptic processing of biopharmaceuticals. And in October, we will have our Asia Pacific Regulatory Conference also in Singapore. So I hope you will check our website out and consider participating in and more of these conferences. When I talk to people about PDA, I think it's really important to stress that PDA is a member-based organization and a volunteer-based organization. Long before I joined PDA as the president, I was a very active volunteer. And every year we have more than 2,500 people who volunteer in one form or another to participate in PDA. And people ask me, well, how do I get started? And so I've sharing this with you so that you understand that there are many ways that you can get started. You can contribute an article to the letter or posting to one of our platforms. You could write a scientific article and submit it to our journal, and so on. So there are many ways for you to get involved, up to and including in leadership roles in the organization. So that's great, but what could you do? Well, we don't have a chapter in Indonesia now, but you can be active in helping us establish one. And you could become active in our Singapore chapter. When we have a chapter, you can speak at a chapter meeting. That's usually very accessible. It's an excellent opportunity to get started. You can volunteer to our Asia Pacific team to be on a conference planning committee. You can write an article for the PDA letter. We publish the PDA letter continuously, so there are many opportunities to publish articles about a wide variety of topics. We also have several times a year, we are doing an Asia Pacific focus edition. So there's another opportunity. We do have a peer-reviewed scientific journal, the PDA Journal of Pharmaceutical Science and Technology. And if you have research or a scientific paper that you would like to submit to a global audience, you can submit it. It will be peer-reviewed and go through our editorial process. If you have the capability and the expertise, you could volunteer to give a presentation at one of our global conferences. We do about 20 to 30 per year around the world. We have a lot of PDA technical reports, but they're all written in English. We have had a lot of interest from some of our volunteers who are bilingual to work with us to translate some of those technical reports into other languages. We do have a process for that. So if that would be something you would be interested in, there's another opportunity to volunteer. You may not be ready to be an expert on one of our technical documents, but we welcome people to become peer reviewers. We need people at different levels of expertise to review our draft technical documents so that we can be sure that they are understandable by people across the spectrum. You can volunteer to be a member of a PDA task force. At any given time, we have about 50 task forces. So every technical report, every standard, every points to consider, every regulatory comment that PDA generates is generated by a task force made up of volunteers. And again, we do comment on a lot of regulatory documents. If you're interested and have expertise, you can volunteer to be part of one of those. So there are many ways for you to get involved. How do you get started? You know, if you know me, you can send me an email. I get lots of emails from people that I don't know. They say, I want to be involved, and I try to connect them. But you can also contact your local PDA chapter, or in your case, you could contact our Singapore office, and I will have information at the end of my presentation about that. There is an email address, volunteer@pda.org. You can go online, you can fill out a little form, and that will give you an opportunity to state what you want to do. You could also contact our PDA letter editor directly and say, I've got an idea for an article. The same way you can submit through our editorial process an article to our journal, you can contact us and say, you know, I would be very interested in speaking on a topic at one of your global conferences, and so on. So there are a lot of opportunities. the simplest way to volunteer is to go to volunteer, send an email, go to volunteer@pda.org, fill out a little form, and let us know what you would like to do. But I would also urge you to contact our Asia Pacific office. They're in your time zone. They can help you with this as well. Since we don't have a chapter in Indonesia, I thought it would be good to share with you some guidance on how to establish a new chapter. We have, in the time that I've been president, the last 11 years, we have created a number of new chapters. So first and foremost, we have to have members in whatever region we are talking about establishing a chapter. So in Indonesia, for example, we could establish a chapter. Generally speaking, you need to have about 100 members in that region before it's sustainable to create a chapter. At the time that you do have that, then you can get together. There's a form we can help you with, but you have to identify members who will agree to be volunteer leaders, and those leaders should be open to any participant who's a member, and should be diverse. It should be from different types of organizations, manufacturing, pharma companies, biotech, suppliers, consultants, health authorities. We are very open to participation. And then if you are interested, if you get to that point, someone from our membership team will work with you, and they will help you to fill out the form so that there's a petition that would have to go to our board of directors, and they would help you with that. And then the board of directors would consider that and decide whether or not to grant you a new chapter, in which case then we would work with you to establish the necessary procedures and goals for you to do that. PDA would work with the new chapter to help you get started, as we have done in Singapore and India and in many other new chapters around the world. As I mentioned, PDA is very well known for our technical documents. If you have not ever had the opportunity to see a PDA technical report, I would urge you to consider that. We have a number. These are just some recent ones that are published, and it's probably not even up to date. I think we've had a couple since then, but you can see they cover a wide variety of topics, in integrating data integrity requirements into manufacturing and packaging, virus contamination in biomanufacturing, low endotoxin recovery, cell-based therapy control strategy. We have a technical report that was translated into Spanish. We also develop points to consider on a wide variety of topics. And we do research, and we publish the results of that research, as you can see. So if you are a member of PDA, every time we publish a document, you have the opportunity to download it for free. If you are not a member, you are free to purchase these documents. And all members have access to a portal where you can view them all the time at no charge. So that was really designed to talk about PDA. In my remaining few minutes, I want to just share with you some of the current issues in pharmaceutical manufacturing, especially in light of the global coronavirus pandemic. And I'm sure many of these issues are going to be very familiar with you. So unfortunately, yesterday I updated this. Since about a year ago, since the announcement of a global pandemic, we have had over 119 million cases worldwide, and more than 2.6 million people have died. In Indonesia, as you can see, we have had a very significant number, but fortunately not nearly as many as in some other countries around the world. But this is truly a global event. I talk to people all around the world. You cannot find a place in the world where they have not been impacted by this global pandemic. As this issue began to evolve from a pharmaceutical manufacturing perspective, some of the first issues that we had to deal with were, how do we stay in production? You know, our employees are susceptible to being infected by this virus just like anybody else. So the short term, there was very clear issues about how do we stay in production. Then there were supply chain issues, and we had to deal with those, both from a perspective of staying in operation and also providing the products that were needed for treatment of people who had developed this disease. And of course, very rapidly, our industry had to start focusing on developing and evaluating therapies to mitigate the effects of this disease in people who become infected. That activity is still continuing. And of course, an unprecedented effort worldwide to develop and evaluate vaccines. That effort has been unprecedented in being successful in developing some vaccines that are now available in different countries around the world. But the new challenge is scale up and distribution of these vaccines to the whole world. Of course, it's very important to maintain production. Medicines are critical every day. If you think about especially parenteral medications, anesthetics, antibiotics, and so on, people need these. These are critical. So you have to continue manufacturing these products. And so pharma companies had to figure out what they needed to do to protect their workers so that they could continue to manufacture. And I can tell you, for PDA and for many companies around the world, all non-essential staff have been working remotely since March of last year. However, production workers and people involved in testing of these products, they don't have that luxury. They still need to go to work. So that was a critical thing, and we had to deal with that. And of course, we have to be mindful that we don't want to contaminate any of our products with the virus. You know, the supply chain issues became apparent very quickly, and they continue to be an issue. So, you know, very early on, it did not take very long for supply chain disruptions to have a direct impact on manufacturing in pharmaceutical companies. And in some cases, it's continuing to do so. Companies had to react very quickly, and in some cases, had to work together with regulators to make sure that they could continue to supply. The initial impact was on personal protective equipment. There was a dramatic increase in demand and impact on healthcare workers. So, you know, I can't speak for Indonesia, but in the United States, masks were not very common prior to this, except in healthcare settings. And as soon as the pandemic became known, there was a period where you could not find masks, and this became a key issue, not just for the public, but also for manufacturers who needed to supply these materials so that their production workers could continue. The second key thing was the urgent need to develop diagnostics, and this caused a lot of challenges in developing accurate diagnostics, but then also rapidly scaling up the manufacture of those so that they could be deployed, so that we could start to get a better idea of who's infected so that they could be quarantined from other people. And finally, we started to see a very dramatic increase in demand for ancillary drugs that were needed for, especially for people that have respiratory failure, which is one of the primary symptoms of this viral infection. And so anesthetics and antibiotics were critical, and in some cases, those became in very short supply. One of the things that this situation has revealed is the fragility of our global supply chain. And so while we've been reacting to that, certainly within the industry and within PDA, we have all been very mindful that we need to think about how we're managing our supply chains in the future so that we can be resilient and responsible in responding to situations like this. So we've been dealing with the situation, but I think there will be long-term changes that companies will be thinking about in terms of redundant supply, distributed supply, so that in the event of future problems like this, that we won't be quite so vulnerable. There have been drug shortages. So there have been a number of products that have been in drug shortage, especially early on in the pandemic. This resulted from increased demand for the products that were necessary for people that had to be hospitalized. But as production shifted to meet those demands, that created some back orders for other products. And so there was a ripple effect. There was also disruption to the supply chain due to absenteeism of production and testing personnel. And so again, there were impacts based upon the staff of these organizations. And more recently, there have been some issues with imposition of trade barriers to distribution of pharmaceutical products, and I should point out, to supplies that are needed in the manufacturing. So even today, I can tell you there are certain components that are needed for the manufacturing of biologic products that are in very short supply because they are critical to the manufacture of these vaccines, but they're also needed for other products. And so as governments and suppliers have had to prioritize to the vaccine production, that has created some shortages in other areas. There have been shortages in a number of different products, as I mentioned, antibiotics, sedatives, those things that are used for people that are on ventilators or with severe respiratory distress. Of course, the regulators have had to deal with this situation too. And very early in 2020, all regulatory authorities around the world had to start taking action. Many of them suspended travel for inspectors early in this process, and in some cases have continued to suspend travel for inspectors. They have accelerated the review of diagnostics, therapies, and now vaccines, but that has also put a challenge on the review of other products. They have been very active in focusing on shortages that are created because of this, and that has sometimes required them to consider emergency use authorizations for importation of products that maybe weren't otherwise approved, but because of the shortage, they need to allow products from other locations. And of course, as we start getting into the new therapies and the vaccines, those have been allowed. And in fact, in the United States and Europe and in many countries, all of the vaccines are only available currently under emergency use authorization. The regulators around the world have also been very active in publishing guidance, and I am very pleased to say they have been very engaged with PDA in sharing information with us so that we can communicate it to our community. Of course, one of the things that I think our industry can be very, very proud of is how rapidly we came together to start working on developing therapies and vaccines. So, of course, the very first thing that was started was developing therapies. There are antiviral drugs that have been available for certain other viruses. So early on, there were a lot of studies to try to determine which of those antiviral products might also be effective against this particular virus. The same with antibodies, either monoclonal antibodies or antibodies from people who have recovered. That is still being explored. There are plasma therapies, immune response modifiers. And so around the world, there have been a number of therapies that have been approved in different markets that have had some level of effectiveness at mitigating the effects of this virus. Of course, there's been tremendous focus on developing vaccines. And, I've been in this industry a long time. I have never seen the level of focus and activity developing vaccines, and not just the traditional, such as the whole virus vaccines, both inactivated and live attenuated, but also genetically modified vaccines, RNA vaccines, viral vector vaccines, protein-based vaccines, recombinant vaccines. So a number of different ways in which you could approach the development of a vaccine for this particular virus have been developed. And as you know by now, a number of these have now reached the stage where they are available for emergency use in different markets. And there are others that are continuing to be evaluated. So at least in the US, I can tell you, we now currently have 3 vaccines that have been approved for emergency use, and that has dramatically expanded the accessibility of vaccine to our population. And I think the same thing is happening in other countries around the world. But, once a product has been identified and proven to be acceptable, that's in some ways that maybe is the easy part. Because getting it, as this headline said, to tens of millions, it's really tens of billions. So, worldwide, we're going to have to provide billions of doses of these vaccines to protect the world for this. Some of these vaccines require multiple doses, so that just doubles the number that are going to be needed. Adding 10 billion doses of any parenteral product in 1 year is a dramatic increase. So everything that you can think of, from the number of components that are needed, the manufacturing effort, the distribution, the marshaling of resources to be able to get these products properly stored to places where they can be administered, organizing to administer these products to a global community. These are very challenging issues and can only be done through a cooperative effort between industry and governments, and that's what's happening. Of course, the rest of the world hasn't stopped because of COVID, and there have been a number of other things, and I'll just briefly touch on a couple of those. Some of this is very much related to the coronavirus situation, but even before this, there have been a lot of efforts focusing on regulatory collaboration regarding inspections. There are a number of different mutual recognition agreements between different health authorities. PICS membership has been expanding, and that has created a way in which inspectorates can gain confidence in each other's inspections. There are some regional alliances around the world. So all of these efforts are focused on increasing the amount of regulatory collaboration, especially when it is regard to inspections. And there are some specific ones on sterile dosage form. As you can see, there's a sterile finished drug product inspection pilot that's going on between a number of different regulatory bodies, and that is underway. It's a little uncertain to me whether they're going to have to change the timing of that because of the global pandemic. But the goal there was to have routine surveillance inspections of GMP compliant sites with a group of these inspectors, so that in the future they would be able to mutually recognize each other's inspections. And from an industry standpoint, I think we're all hopeful that that would be successful. If you haven't heard the term advanced therapy medicinal product, you will certainly be hearing about it. This general category covers a pretty wide variety of products. The term ATMP is predominantly originated in Europe. In the United States, it was more common to use the terms cell therapy or gene therapy, but increasingly the term ATMP is being used. And so these products are at the forefront of medical development right now. PDA has been involved in this for more than 10 years. We established an advisory board on ATMP last year. We will be doing a couple of conferences this year. And as you saw, we have technical reports, but I can tell you that there's a lot of development activity going on in this area for the treatment of otherwise untreatable diseases. And so these products require different approaches to manufacturing and control, and PDA is involved in that. And finally, I'm sure everyone is familiar with Annex 1. This is the EU guidance around manufacturing of sterile products. This has also got a parallel document within PICS, and this has been under revision for the last couple of years. PDA has been very involved in reviewing each version of this revision as it's come out. And in fact, we are continuing to interact with regulators. Even today, we sent a combined comment from a number of different industry associations to the EMA, and we are offering to continue to stay engaged with them. We will have to wait and see when they will publish the final version of that document, but we expect it to be before the end of the year. As I mentioned, we have been active with a number of different organizations in working together to try to come up with some common feedback to provide to the regulators. And I'm glad to say that we've been able to do that. Of course, aseptic manufacturing is one of the most complicated types of manufacturing because it's not a single process. It's a collection of processes. And all of these processes have to be very carefully controlled in order to achieve the desired result, which is a sterile product at the end. And so, again, PDA is very involved in this area. If I look at this chart, almost every element on this chart, PDA has technical guidance and training on those topics. So I've covered a number of things. Hopefully I haven't run over time. You know, I would just like to leave you with the fact that the global pandemic has demonstrated the strengths, but also some of the weaknesses in our global supply chain. The only way we can move forward is the collaboration between governments, the industry, healthcare providers, and a larger community of nonprofits, suppliers, and others. We need to resolve this pandemic and apply the lessons learned so that we can be better able to react in the next one. And let's remember that ultimately our goal is to help all those millions of people around the world that have been impacted directly and indirectly and who are depending on us to solve these problems. Thank you very much.
Thank you very much, Mr. Richard Johnson, for the very insightful talk and for sharing your knowledge and experience with us. Now, I would like to open the Q&A session. For the rest, you can deliver your Q&A, and if you have any questions, you can deliver it through the Google form or through the Q&A box in Zoom. However, since we're a bit short on time, I probably will pick the questions to be answered by Mr. Richard M. Johnson. So for Mr. Richard M. Johnson, just maybe one quick question before we move on to the next session. What do you think the support that PDA can offer and give for countries that are planning to establish a chapter in their countries? What are the highlights of what PDA support that can be retrieved or achieved by these countries? Thank you.
Yes, thank you for the question. So ultimately, the reason why people in a country such as Indonesia would want to establish a chapter is to be able to access the kind of information that PDA members can get. That information, that technical knowledge is invaluable if you are trying to manufacture product of the highest quality, not just for the local market, but for the global market. So when I talk to people in other parts of the world, and by the way, I've been to Indonesia many times. Early in my career, I actually built a manufacturing plant in Indonesia. So I can tell you that when I talk to people in the countries that are developing and expanding their pharmaceutical activities, one of the first things that they ask for is training. And PDA, through the auspices of a chapter, we can work with you to provide the kind of training that you need. We can provide access to technical guidance that would help you to interpret what is the regulator, what are the regulatory requirements, but more importantly, what are the best practices? How do you really do these things? If you look at a regulation, it gives you what you have to achieve, but it doesn't tell you how to do it. And that's really the space that PDA can help with. Because the people who work on these things at PDA, people like myself, I've been doing this for a long time. It's practical knowledge. And you learn from people just like yourselves that are doing the same things that you're trying to do. So I would hope that people in a place like Indonesia that are thinking about establishing a chapter would say, we want to have access to the same body of knowledge, and ultimately we'd like to contribute to it and become a partner in it. And so if that's something that's of interest to you, that is something that we would be very happy to work with you to help you achieve that.
Okay, thank you very much, Mr. Richard M. Johnson, for the answer to the questions and also for sharing the talk. I would like to end the first session. Again, thank you very much. We are very honored to have you here giving your talk to us and sharing your experience and knowledge. And now,
Yes.
thank you.
Thank you. And I will leave. So
Yeah.
Thank you all. Bye-bye.
Bye. Goodbye, Richard. Thank you very much. Okay, audiences, now we will move on to the next session. We will be having a talk by Mr. John G. Strabochnik, PhD. Now, before we begin the talk, I would like to give a brief introduction. So Mr. John G. Strabochnik is the principal consultant from Insight Pharma Consulting LLS. Today, we will be giving the talk with the topic Visual Inspection of Injectables Product, What's Changing and What's Not. Mr. John G. Strabochnik, PhD, graduated from Indiana University, majoring in analytical chemistry for his PhD, and he finished his bachelor degree from Carroll College, majoring in chemistry. He is the founder of Insight Pharma Consulting, and he had experiences as managing microbiology and aseptic manufacturing technical support for Pfizer's global quality operations. He was also the director of the Center for Advanced Sterile Technology and also the director of technical support and engineering for Pharmacia. He was also the process analytical technology and visual inspection expert in the Upjohn Company. And currently, he is also affiliated as an active member and instructor of PDA's Training and Research Institute, and he served as a chair of Board of Directors and chair of the Science Advisory Board. He also organizes and leads the visual inspection internet group, and he's also serves on the USP dosage forms expert committee. He also acts as the chairs of the visual inspection of parenterals expert panels, and he has published and co-authored a book with the title of Visual Inspection and Particulate Control. Mr. John G. Srebosnik, PhD, we would like to invite you to give your presentation. Thank you very much. The time is yours.
Thank you very much, and thank you for the kind introduction. Let me just share my screen, and I can begin. And there we go. Again, thank you for the kind introduction. It's really a pleasure to join you and your colleagues in Indonesia today. I'm very excited to talk about one of my favorite subjects, visual inspection. And I'm also want to share my thanks to PDA for the invitation to be with you today. Before I start talking about visual inspection and detecting and rejecting defective units, I always want to talk just briefly about prevention. And sometimes we get so focused on the topic of visual inspection that we forget about using what we learned from our visual inspection program to prevent defects. The best defect, if you will, is the one that we prevent, the one that never occurs. And so we want to take what we learned from our visual inspection program and make sure we apply it to improving our manufacturing process. We talk about this in terms of a life cycle and the idea of continuous process improvement. I'll also spend a lot of time talking about inspecting for particles. And if you notice, most of the guidances, most of the pharmacopoeias talk very much about particles. But please don't forget about the other visible defects that should be found during your visual inspection process, especially those associated with container integrity, because frankly, these can pose a greater risk to patients than those from particles. So with that, let's move on. The agenda of topics that I want to speak about today, Richard gave a wonderful talk, really talking about the global pandemic and really the response of our industry. And I think that's a response we can all be very proud of. I want to take just one slide also to talk about the global pandemic, specifically with respect to the visual inspection programs. I want to talk about the current regulatory environment, FDA guidance and recalls, the US Pharmacopoeia particulate chapters, the regulations in Europe, both from EMA and from the European Pharmacopoeia. Talk a little bit about some exciting new automated visual inspection technologies, wrap things up with a summary and conclusions, and then allow some time for questions and answers at the end. So first of all, the global pandemic. That's obviously the topic in front of all of us today. It's affecting all of our lives directly, and it's affecting what we do as an industry. And as I mentioned, I'm very proud of our response as an industry to this, and I'm optimistic going forward as we start to see improvements around the globe. Clearly, visual inspection continues to play an important role in the development and delivery of safe and effective medicines. And that certainly includes those associated with either vaccines or for treatments for COVID. And so we are very much in the middle of this when we're talking about visual inspection. Visual inspection can be on the critical path for manufacturing. There are times when the capacity of the visual inspection process will limit the amount of product that can be delivered to the marketplace. So we have to look at the VI process very carefully. Large demand and production volumes, and Richard was talking about some absolutely huge numbers, hundreds of billions of units that will need to be inspected. These large demands should be anticipated. I think the other important thing to think about is you should not expect reduced quality standards or lowered expectations for these COVID vaccines or COVID treatments. In other words, it's not that because of this global pandemic, the regulators or we as manufacturers should expect less in terms of quality and specifically with our visual inspection programs. We should expect to do what we are doing today for all of the other vaccine products or other therapies, injectable therapies that are currently being manufactured. So with that in mind, start early. The lead time for training inspectors, for purchasing automated inspection equipment can be long, so we should anticipate that, making sure that, again, visual inspection does not end up being an obstacle to rapid delivery of these critical vaccines and therapies. So again, start early, but anticipate that you're going to need to do what you are doing now for your other injectable products. So let's look at that current regulatory environment. If we look at three of the major pharmacopoeias, the US pharmacopoeia, the European pharmacopoeia, and the Japanese pharmacopoeia, and referenced at the top are the chapters in each for visible inspection or visible particulates, I'm happy to report that in recent years, we have achieved informally a harmonization of the inspection criteria. And these are reference criteria. You are free to use other inspection conditions, but you should demonstrate that if you do something different than this, that what you're doing is as good as or better than what's found in the pharmacopoeias. You can see here light intensities, 2000 to 3750 lux, higher intensities, especially referenced in the Japanese pharmacopoeia when you're looking at plastic containers or some other difficult to inspect products. An inspection time of 10 seconds, and that is against both a black and a white background. 5 seconds against one, 5 seconds against the other. We need contrast to see the various particles that we're looking for. Some may be light in color, some may be dark. So by using both black and white backgrounds, we achieve good contrast and good sensitivity for a wide range of particles that may be found in the products we're manufacturing. When it comes to acceptance criteria, there are still some differences. In the US, we have, for many years, had an acceptance criteria of essentially free from visible particulates. More recently, we have added a numerical specification based on the widely used sampling plans, AQLs, that we use for these products, establishing an AQL of 0.65 to be associated with particulates. In Europe, the expectation is clear and practically particle-free, and in Japan, free of readily detectable foreign insoluble matter. Again, all of those recognize that even the best inspection method, the best human inspector, the best machine we can buy today is not perfect in its inspection ability. We have to do the best job that we can, but it recognizes that there is not an absolute certainty in the removal of all particles of all types. And so because of that probabilistic nature of the inspection, there is some then flexibility in these acceptance criteria. Let's look a little bit more at what we mean when we talk about visible particles, because that's the next question. None of these chapters, none of these guides in the pharmacopoeias tell you exactly what size particle you need to see. However, today there is general agreement that the visible range starts at 100 microns. That's, if you look at the average human hair, about the diameter of that hair, or if you take a pen and make a small dot on a piece of paper, that's about the diameter of the dot that you would make. So these are small, but visible. And generally, we're talking about things bigger than this in the visible range. How do we detect those? With the human eye through manual human inspection, semi-automated inspection, where we use a machine to spin, to rotate the vials, to hold them in front of the inspector, but there's still a human inspector, their eye and their brain that decides whether it's good or bad. Or we can use fully automated systems where we use cameras, computer algorithms in order to assess whether a given vial, syringe, ampule meets our acceptance criteria. We also talk about subvisible particles, and generally these are between 1 and 100 microns. They are generally outside of the range that we can see reliably with the human eye. And so we use instrumental methods, light obscuration, microscopy, flow microscopy, and an older electrical method, Coulter counters. There are also much smaller particles, the nanometer and submicron range, where we use laboratory instrumental methods to assess these. Generally, we do not assess these as part of manufacturing and batch release, but rather during development, developing formulations to minimize these very tiny particles. We can also talk about general particulate types based on where they come from. And we talk about extrinsic particles. Those are from outside the process. They're uncontrolled. They're generally environmental contaminants. And as you see with the risk arrow on the side of your screen, they are generally assigned the greatest risk because we don't know where they came from. And as a result, we don't know their microbial load. Things like insect parts, hair, fibers, paint, rust are examples of these. So they may carry microorganisms and affect the sterility of the product. And we also don't know whether our product is stable in contact with these things. And so it might affect the stability of the product as well. Another category of particles are those that we call intrinsic. Those come from within the process. They're unplanned. We don't want them in our products, but we do see them generally at very low levels. These are generally associated with the processing equipment or the primary package, but they are generally qualified product contact materials and are generally coming from sterilized or at least sanitized components. Things like stainless steel, glass, rubber, silicone oil. So they pose a lesser risk to both to the patient and to the product stability itself. And there's a third category. We call these inherent. These are particles that are actually part of the formulation. They need to be controlled, but they are expected. A real easy example is if we are making a product that is a suspension, then we have drug substance that is part of that formulation that is there in particle form. So we want those particles to be present. We may add adjuvants to vaccine products that remain undissolved in particle form as well. When we make protein solutions, some of those large molecules may agglomerate and form visible protein agglomerates. While not desirable, it may be acceptable at low levels. This acceptability, however, has to be demonstrated through your clinical studies. So again, this is why it needs to be controlled, but it is not unexpected. What can we expect from human inspection? And obviously, this is dependent on the particle characteristics as well as the product and package characteristics. The example I'm showing here is in a clear 10 ml tubing glass vial in water for injection, and these are single particles of increasing size. And what you can see is at 50 microns, that's a threshold of visibility, but with a very low probability of detection. At 100 microns, where we say the visible range begins, we have roughly 40–50% probability of detection. So we certainly do not see every 100 micron particle, but we will see some of them. When we get to about 150 microns, we cross the 70% threshold. This is the threshold that Jules Knapp defined as the reject zone threshold, and things with a probability of detection of 70% above, we now start to say are more reliably detected. And we must get up to about 200 microns before we see them being detected at over 95 or 99% probability. There's always still some uncertainty, and I'm hesitant to even say 100% at that point, but we have a high probability of detecting particles 200 microns or above. But again, these are specific particles. These happen to be glass and polystyrene particles, spherical in shape, in clear liquid, in a clear vial. Generally, larger vials, colored solutions, colored vials, more viscous solutions, all of these things will make detection more difficult and shift this curve to higher detection numbers, higher particle sizes before we cross that 70% threshold. What can we expect from a human inspector? We need to control their visual acuity. We want to do regular annual eye checks to make sure the equipment is working as intended. People get tired, and with fatigue, performance degrades. So we need to give frequent breaks in order to maintain reliable performance, and we don't want to inspect for too many hours in a day. So generally, we limit it to a single 8-hour day or 8-hour shift, and we inspect for no more than 1 hour before giving a short break, maybe 5 minutes, or it might be a lunch break or a tea break, something to refocus the eyes, refocus the mind. But people have one amazing property, and that's flexibility. Even if they have not seen something before, they may say, this looks wrong, this looks different, I have not seen something that looks like this, and they should talk to their supervisor and react to that. That's something that machines may not always be able to do. But inspection is probabilistic. And that means that even though we do 100% inspection, we look at every container, we will not see every defect 100% of the time. And as you saw in that earlier graph, the probability is associated in part with the size of the particle. Smaller particles are harder to see than larger particles. This is a few things that I've excerpted from a talk by Ruckman de Silva, and he is part of CEDAR at US FDA. He gave a nice summary of FDA's thoughts on visual inspection at our PDA Visual Inspection Interest Group workshop held last fall. And he talked about expectations during development as well as expectations upon approval. And so to start with, the point is you need to characterize visible particulates at release, during stability, and during and after exposure to in-use conditions. So you need to be thinking about particles throughout the product life cycle, from formulation, sitting on the shelf, during use. You want to have strategies implemented to minimize visible particles in the formulation during development. And certainly you're going to minimize them during manufacture as well by following good manufacturing practices. Generally, the USP recommended particulate acceptance criteria and inspection conditions that are found in chapters 1 and 790 are the ones that FDA looks to for release and stability specifications. If you find visible particles, you should assess their risk to understand the impact on product safety. So that might, as we were talking about extrinsic, intrinsic, and inherent particles, knowing more about what the particle is, its size, where it came from, can help you assess that risk. You want to then develop strategies to mitigate that risk, ideally prevent those particles from entering into the finished product. And you also want to look at trends. If you see an unusual trend during storage, you want to investigate to identify the root cause in order to, again, have further mitigation strategies. Now, upon approval, you also need to have adequate visible particulate characterization and control strategies. So again, good environmental controls, good cleaning processes for your manufacturing equipment, good cleaning, good selection of your primary packaging materials, the stoppers, the vials, the syringes, the plungers that are used, good filtration during filling, good environmental controls, as I mentioned. All of these things lead to a very low particle burden in the finished product. And again, pointing to USP 1 and 790, setting the minimum requirements for inspection and acceptance of products with regard to visible particulates. And you should have good analytical methods that are validated, qualified in order to detect and quantify visible particulates. And this would extend as well to subvisible particles in your solution products, and also microscopic methods that you might use to measure and identify particles in the laboratory as part of an investigation. A few other useful things from US FDA. They have a compliance program guidance manual, this long string of numbers that you see here that they issued in 2012. This is a guidance that they wrote for themselves to train their own inspectors when they come to a plant that makes sterile injectable products. There are many topics covered, but there is a good section that covers the visual inspection program. So it's nice to see what they are looking for when they come to your plant. I can very briefly summarize that they are most concerned with particles and container integrity, cracks. And so those are of great concern. Inspector training is of great concern, and your investigations. How do you detect and respond to these critical defects when you find them? There are other details that are there, but I think those are the ones that really stand out to me as very important to remember. FDA also has a planned guidance. They've been writing this for a number of years. It's called Inspection of Injectable Products for Visible Particulates. The best information that I have is that we may see this guidance this year. I understand that it was finished last year, going through internal review at FDA. So I hope that we will see it sometime this year, and it will provide, again, further guidance for our industry on this very important topic. If we look at FDA recall notices over this 5-year period, essentially the last 5 years, you'll notice that the majority of those, or at least the largest number of those recalls, 38%, were associated with visible particles. So that's why we talk about visible particles so much. But almost an equal number were associated with the lack of sterility assurance. And you would not be surprised that these are two of the more predominant failure modes when making sterile injectable products. Other recall reasons: labeling, containers, others, and you can see the examples of others, whether it's incorrect potency, discoloration, impurities, degradation, storage temperature excursion, a wide range of other things that are also associated. But again, when FDA, other regulatory authorities see such large numbers of recalls associated with one thing, visible particles, they are going to look hard at the visual inspection process. Let's look at these recalls, first of all, just by therapeutic area. And you'll see that it goes up and down from year to year. If we look at the most recent year, 2020, what's interesting is you see that big red bar. A very large number of recalls were associated with topical products, which in past years is quite unusual. This is pandemic driven. It was problems with unapproved hand sanitizers. And so a large amount of that FDA activity was focused on those recalls. You can see that there was also a fair number of recalls in the last few years associated with solid oral dosage forms. Many of these were associated with impurities in some of the oral products, either for diabetes or for, excuse me, for acid reflux. And so again, these were causing significant numbers of recalls. You look at the blue lines on the bottom, those are the ones associated with parenteral products. And I'm going to look at those in more detail right now. And you can see that that number peaked in the year 2014. That peak was driven in part by a number of very, no pun intended, visible recalls associated with glass and metal particles in the preceding years. That sensitized the industry as well as regulators to look harder, and the more you look, the more you find. So more lots of product were recalled as a result of visible particles. I'm happy to report in 2014, the USP published General Chapter 790, which provided more direct guidance on visible particle inspection and acceptance criteria. I think that helped in part to set clear guidance and then bring numbers back down more in the historical range. On average, we see about 10 recalls per year. That's not a good number. I think as an industry, we can do better, but there are still opportunities for improvement. You can also see in 2020, that's the lowest number that we've seen in a long time. I'd like to think that's because we've gotten so much better and we can pat ourselves on the back and say we've done a very good job. I think it's also true that a lot more attention was being placed on preparation for vaccines and therapies for pandemic treatment, and therefore not as much focus was placed on visual inspection programs. So I don't think we should be complacent. We still have work to do. These are other chapters in the USP that are associated with particulates. I've highlighted 790 and 1790. 1790 is the information chapter that gives further guidance on how to perform what is provided in the required chapter of 790. You'll see that there are other chapters that apply to subvisible rather than visible particles, or that provide additional focus on therapeutic areas like ophthalmic solutions or therapeutic proteins. All of these either are required if you're making that particular kind of product, or chapters with numbers greater than 1,000 provide guidance. They are information chapters. 790, as I mentioned, provides the inspection conditions and lot acceptance criteria. It also talks about doing supplemental testing for difficult to inspect products, DIP products. And these are things like freeze-dried products, where you take a small number and perform a destructive test. You dissolve the freeze-dried cake and look for particles in the resulting solution. And so there's guidance on that topic there, as well as more guidance in USP 1790. 790 became official in August of 2014. 1790, the information chapter, to help you comply with what's in 790, became official August of 2017. We are in the process, and I lead the team at USP who is working on this to revise 1790, and I'll tell you a little bit about what changes will be coming. A revised draft was published last November in the Pharmacopeal Forum, and I hope that a new draft, rather not a draft, but a final version will be published sometime mid-year of this year. So what's in 1790? It talks about the typical inspection process flow, the idea of doing 100% inspection, taking a sample of those that are inspected, and then inspecting again, doing an AQL inspection or acceptance sampling. Talks about the acceptance life cycle that I mentioned earlier, the idea of continuous process improvement, looking at inspection results to improve the inspection process. Talks about how to interpret your inspection results. Talks about different inspection methods and technologies, manual inspection, semi-automated, automated inspection. Talks about how to qualify human inspectors or validate automated inspection equipment. Talks about what to do if I have complaints about particles in product that I've released and is in the marketplace. And finally goes through further conclusions and recommendations, provides a long list of references as to why these particular guidance have been given. What's changing? We are updating the sections on difficult to inspect products. Richard talked about ATMPs. We wanted to give more guidance on inspection methods and expectations for cell and gene therapy. We want to talk more about and give better guidance on ophthalmics and also product packaged in opaque plastic containers or flexible bags. These are difficult to inspect. We also expanded discussions for alternative sampling plans, especially when you have very small batch sizes. The traditional sampling plans may not be the best or easiest to use. We talk a bit, and I'll talk a bit more today, about the use of artificial intelligence and deep learning to hopefully improve our ability to do automated visual inspection. And then we updated the references because there have been some good publication since the original publication of this chapter. In Europe, there are also good work that is being done. Richard talked about EMA Annex 1, and there have been an expansion of the discussion on visual inspection in Annex 1. And so again, I would refer you to the published drafts in 2017 and 2020. We are hoping that we will see the official final version sometime this year, as Richard mentioned. There is quite a bit of discussion, but I will say there is generally good harmonization, what you find in Annex 1 and what has already been published in USP Information Chapter 1790. I don't find them in conflict. I think sometimes they are making different points, but in general, they are complementary to each other. When we look at the European Pharmacopoeia, the chapter that talks about visible inspection, visible particulates, is 2.9.20. And minor revisions were made to that chapter, and those changes became official in January of 2020. So those changes are now in effect. Those changes, I'm going to summarize in just a moment. I'm sorry. I want to cover a couple of other points here first. There is a new chapter in the EP 5.17.2. This is an information chapter. It is recommendations. It is a good chapter. It expands on what was in 2.9.20. However, there are some inconsistencies in terminology between what is in the USP and what is now in the EP. They do not use the distinction with regard to inherent particles that I described earlier. They have put those in the intrinsic category and only have extrinsic and intrinsic particles that are defined. So this may cause some confusion. This became official in January of this year. It was published for comment earlier, but now is an official chapter as an information chapter or as a guidance. Don't forget about an older chapter originally published in 2008 for monoclonal antibodies. If you are making monoclonal antibodies, this is a bit in conflict with other European Pharmacopoeia chapters because it sets an absolute expectation for no particles. Not that it is free of visible particles or practically free or essentially free, but it expects these products to be absolutely free. And there's no immediate justification given, but there is an expectation that additional analytical work be done to support the particle load in those particular dosage forms. Now, what was added to 2.9.20? This is broadening the scope from just clear solution to liquid preparations. And these include powders, freeze-dried products that are reconstituted for administration. It adds using LED light sources, light-emitting diodes as suitable light sources. This is becoming a much more common practice. It also recommends the use of increased light levels and longer observation times when you're looking at colored or turbid preparation. colored glass containers, plastic containers. This aligns very nicely what's in the Japanese pharmacopoeia, what's also in the USP. So again, I think we are seeing some good, more harmonized guidance in this area. And it further defines inspection should be performed without magnification, which is true of the other pharmacopoeias as well. And it does say that if the primary container limits inspection, then transferring to another container is recommended. This is that supplemental or destructive testing on a small number of units to confirm that at least in that small number of units, there is no large evidence for particles being present in the batch. So again, general considerations, workflow agree with the recommendations in USP 1790. Definitions of extrinsic, intrinsic particles do not align. So again, this is where there are some differences in 5.12.7. I'm going to wrap up here now talking about some new automated visual inspection technologies. First of all, and you're seeing the application of artificial intelligence or machine learning in many areas, whether it's in the phone that you carry every day, whether it is in our development and our investigational processes as we search for new drug candidates, whether it's facial recognition, these tools are very powerful. And most of them are designed to mimic how the human brain works. And we're taking advantage of that here. You can have either supervised or unsupervised learning. Supervised learning, we have an expert inspector goes through, finds a bunch of defects, and now those are shown to the machine, to a camera, and then the machine can analyze those images and learn what a defective unit looks like. On the other hand, we can have unsupervised learning, and there the machine generally learns what a good vial looks like, and then reacts to anything that doesn't look like those good vials. Both of those are valid methods that are being explored for the visual inspection process for injectable products. The key thing here is that they do not continue to learn either supervised or unsupervised. We go through this learning process and then we freeze that learning, and then we go through a more traditional validation process. So from a compliance standpoint, it behaves much like the other things we do, but it hopefully helps us develop more quickly and a more rugged recipe or model for our inspection process. Again, it's doing the same thing that a human inspector is trained to do: target acquisition to find something and then classify, is that good or bad? We hope through this that we can develop smarter recipes or smarter models that will reduce the number of false rejects that we see. Today, machines can be fooled. A bubble looks like a particle and it's going to be rejected, even though it is not a defect. And there are other very minor anomalies in the appearance of good product that often fool machines into rejecting those units, having higher than desirable false reject rates. As I mentioned, you would follow a traditional validation strategy. And so for those reasons, from a compliance standpoint, we are working within really many of the things that we know today. We're just using some earlier development tools to get us to a good place quicker. There's currently an active PDA task force of which I'm a member. We are developing a points to consider document on machine learning applied to visual inspection of filled injectable product. And this document, we have a draft of that, and we expect to publish it a little bit later this year, contains a description of these learning algorithms, how they work, to give you a tutorial. And then it also talks about the regulatory consideration, the validation tools that and approaches, strategies that can be used with these new learning algorithms. So to finish up, there is clearly a high concern with visible particles and container integrity by global authorities. And you saw those large numbers of recalls associated with visible particles. So this should not be surprising. There is improving alignment of the global regulations, and I think that's very positive. Now, these major pharmacopoeias, US, Europe, have agreed on a common set of inspection conditions. At least we are looking at things the same way. 100% inspection, however, does not guarantee detection of all defects. However, you can still achieve reliable results. This is the probabilistic nature of inspection. Visible inspection is a critical step in delivering safe and effective medicines to patients and in maintaining regulatory compliance. So having a robust visual inspection program is a key part of your manufacturing and quality process. And lastly, the regulations and technologies are constantly evolving. New things are coming. We are learning new things and applying that to what we do. So again, it's important to stay active, listen, learn, to stay current with what is going on. One way to help with that, PDA has the Visual Inspection Forum. This year, it will be a virtual meeting coming up next month. This is a 2-day meeting that we have been organizing for now 20 years that really discusses regulations associated with visual inspection, technology associated with visual inspection, case studies, practical learnings in this area. So I would certainly recommend your participation in this meeting, and you can find more information about this meeting on the PDA website. Richard also talked about training being offered by PDA, and I'm very proud to be an instructor at PDA's Training and Research Institute, and I teach a course on visual inspection. We will be teaching a virtual course in April, and then in November, hopefully we will be back in the classroom, back in the laboratory, and offering a hands-on course on visual inspection in Bethesda. So again, this is a very, I think, important part of what PDA has to offer. And with that, this is my contact information. And I will close. Some of you may be familiar with the game show Wheel of Fortune, and I hope you can see and you will know what you are doing, and you can fill in the missing letters and know that we're doing visible inspection.
Okay, thank you very much, Mr. Richard M. Johnson, for the very insightful talk and for sharing your knowledge with us. We already have several questions here. I'm going to read the one that is in the Zoom Q&A chat box first. The question is from Yuli Agung Prasetyo. I want to ask a question for injection products. For particulate matters, should it be checked in every period of real-time stability study, such as T0, T3, T6, and then until shelf life, or we can define only in T0 and shelf life? Thank you.
Generally, it would depend on the product type. So if it is a solution product that can be inspected non-destructively, then generally you would make that assessment at each of the time points, because that's very important, and especially earlier in development, to know whether you have particle generation, especially stability indicating particles. You may have interaction between the product and the glass delamination, or the formation of glass lamellae, and you would want to know when that is occurring. Now, if it is a destructive test, if it's a freeze-dried product, if it is a frozen product, for instance, then you would probably limit the number of those tests that you would do, and you would not make as many of those kinds of inspections under those circumstances. But when you can, if it's a non-destructive measurement, I would certainly encourage you to do that at each of the available time points.
Okay, thank you very much for the answer, Mr. Richard N. Johnson. There's another question from Dewi Arum Sekti. I'm going to translate this. For injection powders, during the inspection for Chapter 790, let's say a fiber is found, what should be done? Is it okay to perform sample inspection?
So what you are trying to do with a freeze-dried product, since you cannot see inside the cake, if you find in your sample of 20, and the general sample size that's recommended for most commercial batch sizes would be 20, the expectation is you would not find any particles in that small sample. If you find a fiber, generally then the next step is to test additional units to get to a sample size where one positive result is acceptable. In other words, one particle is still permitted. Typically, that is going from a sample size of 20 to, say, a sample size of 80. So you'd look at another 60 units, and then hopefully in that total of 80, you would only find one with a particle in it. That's normally how that's addressed. But again, the expectation is that you really aren't going to find a large number of particles. If indeed you just were unlucky in finding that, 1 in 1,000, then by looking at additional samples, you would answer that question. On the other hand, if in your sample of 20, you find 2, 3, or 4 units that have particles in it, then you really know that that batch has more particles in it than is permissible by the expectation of an AQL of 0.65. And generally, you can't remove all visible particles in something like a freeze-dried product through your 100% visual inspection process.
Okay. Thank you very much for the answer, Mr. Richard Johnson. There's another question. Regarding the visual inspection for suspension, let's say we want to inspect like a dark or colored particulate, how is the current acceptance or development for machine inspection when being used for visual inspection?
It depends very much on the nature of the suspension. There are certainly some suspensions that I will call a light suspension that you can actually see through. It looks like a cloudy liquid. Many vaccines fall into this category. And there you can do a very effective job of seeing particles with automated inspection. It's also possible, depending upon the design of the system, to detect some particles in a heavier suspension that settle to the bottom, especially a dark particle against a white suspension that settles to the bottom. And under those circumstances, you can detect some that are on the bottom. And then depending upon the size, the shape of the container, by spinning the container, some dark, heavy particles can be spun to the outside and also detected. And so the automated inspection equipment continues to get better at being able to detect particles in suspensions. But it is very dependent on your particular product, both the design of the container and the nature of the suspension itself. So what I will always recommend in those situations is prepare samples with known particles in them, and then work with the various automated inspection suppliers and have them test, see what they can do with your product. They can certainly give you very good information about how successful you will be with their equipment.
Okay, thank you very much for the answer, Mr. John. Now, the next question would be, can you tell us the best method or the right method to visually inspect emulsion products? What are the proper way to ensure that the product is really free from foreign particles?
So with an emulsion product, it is very similar to what I was just talking about for a suspension. And I will certainly recommend the following: PDA Technical Report 79, which is a technical report that talks about inspecting difficult to inspect products. Suspensions, emulsions, freeze-dried products, amber vials, flexible bags are all examples of these. And it goes through some recommended methods. Now, if I have an emulsion, first of all, when I do the 100% inspection, if I do a manual inspection, I might place a mirror on the tabletop in the inspection booth and gently raise the vial, looking for particles on the bottom before I disturb them, and then go through my normal inspection process. It improves my ability to detect particles that will have settled to the bottom. Then I do the best inspection that I can, and then I would do a supplemental test, which is what's described in more detail in Technical Report 79. For an emulsion, I might, if I have room in the vial, I might add either a solvent or change the pH to break that emulsion in order to now have a clear liquid and look for particles. Or I might dilute the emulsion to better see particles in that vial. Again, depending upon how much room I have in the vial. If I'm unsuccessful in that method, then I would take the emulsion and I would pass it through a coarse filter or mesh that would hold back any particles in the visible range. I might have a 50 micron mesh, which would hold 100 micron or larger particles. And then under low power magnification, I can look at that screen to see if indeed there were any particles in the visible range in that vial. I'd prefer not to open the vial to do that test because there's always a risk of a false positive of some contamination. So I have to work in a very clean environment. If possible, I'd like to do the things inside the vial that I mentioned first if I'm successful.
Okay, thank you very much, Mr. John. The next question will be from Bapak Sumarno. How should we determine, in this case, the lowest limit of the AVI machine?
So generally, AVI machines are validated against a criteria that says they should be as good as or better than a human inspector. So generally, you will have test sets, known defects, good vials that are inspected multiple times by your manual inspection process, referencing what we were just talking about, what is shown in the various pharmacopoeias. That establishes a baseline, a manual baseline. And then you can inspect those same defect examples, good vials, using your machine and compare the performance between the two. There's further discussion on that topic in USP 1790. I also discussed that topic in a book that I wrote that's available from the PDA bookstore. We also discussed that topic in the course that I mentioned through PDA at the Training and Research Institute. But the key here, in my opinion, is to establish what that manual baseline is and then compare your new method, an alternative method, your automated visual inspection machine against that and show that it does as good or better than that manual baseline.
Okay, thank you very much, Mr. John. Now, since we don't have much time left, probably last question. Regarding the qualification for visual inspector, for a human inspection, is there any appropriate age limit? And if the inspector is wearing glasses, is there any limit or any standard? And if you're using a fully automated machine inspection and then you notice, and then you want to do a re-inspection using human inspection, is it allowed? So let's say you have that is already being rejected by the machine and then you do a re-inspection using human inspection. So what is your opinion on that? Thank you.
So let me answer the first ones about manual inspection, and then because this is sort of a multi-part question. So first of all, there is not an age limit. There is a performance limit. And so we do an eye exam, and you can have correction with glasses like I wear or contact lenses if they're permitted in your manufacturing area. The goal here is that you need to have 20/20 near vision that can be corrected with lenses or without, but there is no age limit, but you need to meet those visual criteria as well as then pass the test. You have to have a good test with standards that shows that you can indeed see the defects that you need. So recognizing an age limit, and certainly our eyes age, high, a person can do the job, then they don't have to stop at a particular age. But we have this built-in check. Annually, we're going to, well, first initially qualify, but then every year we're going to retest the eyes, the performance to make sure that they're still doing a good job. So that's how we control this, but there is no age limit that is placed. There is no, prescription limit other than you have to be able to achieve that equivalent to 20/20 vision, near vision, in order to be an inspector. Now, when we talk about the validation and the AVI, and could you repeat, I'm sorry, could you repeat the last part of that question? I'm sorry,
Yes, so
it's
let's
two-stage inspection. I got it. Two-stage inspection. So the question is, if the machine is rejecting, ejecting units, can I inspect those manually and then accept them? Can I change the decision that the machine made? And you can, but you must do this very carefully. Again, we describe this in more detail in USP 1790 and in my book as well. We call this inspection. So when you do this, you now tune the machine. You actually often raise the sensitivity of the machine a bit so that now it is really looking for good units rather than trying to reject bad units. It is tuned to pass the good units, and then everything else is sort of in an uncertain category. Further analysis is needed to decide whether it's good or bad. You can then do that through critical manual inspection. And this might mean a slightly slower inspection, more critical inspection of each unit. And then after that, before you allow those to be put back in with the ones that the machine said was good, you now have two channels. Machine says is good, the people say are good. Manual inspection is good. The machine, that part of it gets its own AQL, a normal AQL, to say that part of the batch is acceptable. The part that the humans inspected, because there's greater risk, the defects are concentrated in the ones that the people were looking at. And we know that even the best human inspector isn't 100% effective. We use a tightened AQL, a more critical AQL, to assess that part of the batch before we bring them back together. So we need these added controls. And you should be doing this, first of all, after you have fully optimized the machine, done everything you can to reduce the level of false rejects. And you need to understand why you're doing it. Is it because there are air bubbles? And what have you done to minimize this before you just start overriding the decision that the machine has made? You should also expect that any audit regulatory authority is going to ask you a lot of questions about that because there is greater risk in this approach. So you have to show how you've added these additional control steps in order to manage that risk.
Okay, so thank you very much, Mr. John. There's actually still a lot of questions. However, unfortunately, because of the limited time, we probably will send these questions directly to Mr. John so he can answer it, and then we will blast it to all of the participants over here. So once again, thank you very much, Mr. John Bushnick, for sharing your knowledge, your expertise with us. Hopefully we could see each other again in other events. Thank you once again. Thank you.
I look forward to that.
Thank you. Thank you so much. Okay.
Thank you very much.
Bye. Yes, thank you.
Bye.
Bye. So we will be continuing to the next session, which will be our last session. And this session will be delivered by Mr. Surya Septefanus as Pharm APT, which is the site head of PT Global Oncolab Pharma. The topic that he will be delivering today will be the path to adaptability and the new normal. He graduated from Universitas Surabaya for both his pharmacist professional and also pharmacy degree. And he's currently the site head of PT Global Oncolab Pharma, and he was the production, logistic, and technical service in PT Finusol Pharma International, the production managers in PT Kalbe Pharma, logistic manager in PT Kalbe Pharma, and logistic supervisor in PT Kalbe Pharma. So that will be my brief introduction for Mr. Surya. Mr. Surya, you may start your presentation. The time is yours. Thank you.
Okay, thank you, Ms. Petra, for your warm introduction. I will start to share my screen. Wait a sec. And hello and good morning, everyone. So allow me to present in Bahasa because we are currently speaking Bahasa and live in Indonesia. So I write the slides in English, so I hope it can serve both ways. So let's get started. You can see the screen here. Yeah. The title is The Path to Adaptability and the New Normal. So the kunci adalah kata kunci dari kalimat tersebut adalah kemampuan untuk beradaptasi menghadapi situasi pandemi yang tahun kemarin kita rasakan bareng-bareng. Bahkan sampai tahun ini pun juga kita masih rasakan dampaknya. Dan bagaimana kita menghadapi tatanan dunia baru atau yang kita biasa sebut new normal, baik di kehidupan nyata sehari-hari ataupun juga di industri. Ini adalah outline yang akan kita bahas, bagaimana kita menghadapi pandemi dan juga dampaknya, adaptability kita menghadapi pandemi sebagai industri farmasi seperti apa, dan juga next normal sebagai pendukung industri 4.0, kita maju sebagai Pharma 4.0 dan juga Quality 4.0. Nah, tahun 2020 kemarin memang benar-benar menjadi tahun yang sangat roller coaster, ups and down. Di awal tahun kita masih belum merasakan dampak pandemi, tapi sejak 11 Maret tahun 2020 kita rasakan setelah pandemi ini ataupun coronavirus ini dinyatakan sebagai pandemi global, kita benar-benar merasakan banyak hal, terutama di sisi industri. Kita merasakan yang namanya penangguhan di production activity, kemudian juga ada pembatasan pergerakan orang, perpindahan orang, dan yang tadi juga sempat disebutkan mengenai supply chain disruption. Kita juga merasakan dampaknya kelangkaan barang, baik itu bahan baku ataupun bahan penolong. Nah, sebagai salah satu bidang industri juga, pharmaceutical dan juga health market dituntut untuk agile dan juga fleksibel terhadap kondisi seperti ini. Jadi walaupun tetap diminta untuk menghasilkan produk yang berkualitas, memiliki safety yang baik dan juga efikasi yang memadai, kita tetap dituntut untuk agile dan juga dituntut untuk fleksibel menghadapi pandemi COVID-19 kemarin. Nah, flexibility ini juga didukung dengan pendekatan quality risk management. Kita lihat kota-kota kosong, kemudian juga kalau kita berpergian, kita nggak bisa berpergian ke luar negeri, mungkin yang udahan perpanjang visa akhirnya atau perpanjang paspor belum bisa dipakai. Nah, ini adalah dampak yang memang kita rasakan selama masa pandemi kemarin dan juga masih kita rasakan saat ini. Fakta-fakta yang kita dapat selama pandemi ini untuk bidang industri farmasi adalah industri farma ini merupakan industri yang esensial untuk mendukung kehidupan. Jadi walaupun industri yang lain pada saat pandemi kemarin di shutdown, tapi industri farmasi tetap bisa menjalankan aktivitasnya. Kemudian kedua, kita tidak bisa menghasilkan produk dari rumah sebenarnya. Jadi industri farmasi berbeda dengan industri lain, di mana mereka bisa work from home, aktivitas bisnisnya tetap bisa berjalan walaupun mereka bekerja di rumah. Tapi kalau di industri, kita bicara di anak pabrik seperti kita, kita harus menghasilkan produk di tempat yang sudah di-certified oleh regulatori kita. Dan yang nomor 3, karena kita ada bagian industri dan kita menghandle people yang cukup banyak, personal behavior memainkan peranan kunci agar persebaran virus dan juga kontaminasi virus ini bisa ditekan. Kalau dulu paradigmanya adalah manusia mengkontaminasi produk, sekarang paradigmanya menjadi manusia dapat mengkontaminasi produk dan juga mengkontaminasi manusia yang lain karena adanya virus COVID-19 ini. Ini adalah survei yang dilakukan oleh PwC di tahun 2020 kemarin, dampak dari COVID-19 terhadap pharma industry adalah seperti apa, terhadap medical institusi dan juga distribusi dan juga health insurance. Kita lihat bahwa ada positif dan juga negatif impact. Di positif impact-nya tentu demand terhadap barang-barang ataupun produk-produk yang mensupport penanganan COVID itu sangat meningkat. Kita tahu di awal tahun kemarin ada kelangkaan produk-produk tertentu yang mungkin lebih banyak terkait dengan imun booster dan juga vaksin development. Seluruh dunia berlomba-lomba untuk menghasilkan vaksin. Ini adalah positive impact. Sedangkan negatifnya di sisi yang lain, kebutuhan untuk produk-produk chronic disease ini jauh menurun. Dan juga yang juga dirasakan terkait dengan pengembangan produk baru. Di mana pengembangan produk baru yang kita membutuhkan clinical trial, kita membutuhkan BS studi dan lain-lainnya yang membutuhkan masa yang cukup banyak, itu menjadi terhambat. Karena pada saat kemarin ada pembatasan sosial, kita tidak diperkenankan untuk mengumpulkan massa yang dalam jumlah yang banyak. Nah, selain itu juga dari sisi teknologi, internet memainkan peranan penting. Kita sekarang sudah banyak menemukan konsultasi dokter secara online. Kita bisa membeli obat, men-scan resep kita secara online tanpa perlu keluar dari rumah. Sehingga yang negatif impact-nya, pasien-pasien yang kemudian untuk membeli obat biasanya pergi ke apotek, ataupun kalau misalnya dia sakit ke rumah sakit, ini jauh menurun. Sehingga benar-benar kerasa kemarin di tahun 2020, penurunan kebutuhan produk-produk yang tidak terkait dengan COVID ini jauh menurun karena memang pasien pun juga tidak banyak yang pergi ke rumah sakit. Nah, untuk menjaga bisnis continuity ataupun keberlangsungan bisnis kita, sebenarnya ada 3 hal yang perlu kita cermati untuk bisa kita lakukan, yaitu pertama terkait dengan personal protection atau biasa disebut dengan PPE. Ini harus didekatkan ataupun harus didasari dari sisi pendekatan secara risk-based assessment. Apa resiko-resiko terhadap personal protection penyebaran virus ini. Kemudian kedua yang harus ditanggulangi juga adalah terkait dengan supply chain disruption. Dan yang ketiga, di satu sisi kita punya problem challenging terkait dengan COVID, di sisi yang lain kita sebenarnya uang. Sebenarnya drug development dan juga new market opportunities ini muncul nih ketika COVID ini keluar. Nah, ini semua yang menjadi peran kita untuk bisa di industri farmasi menjaga agar bisnis kita tetap berlanjut. Kita masuk ke yang pertama, yaitu adalah mengenai risk-based assessment for personal protection. Sebenarnya di industri farmasi sendiri, kita sudah paham benar bahwa pakai masker, kemudian juga sanitasi hygiene perorangan, cuci tangan, itu sebenarnya bukan hal yang baru bagi kita. Tapi dengan adanya pandemi kemarin, memang kita seperti diingatkan kembali, recommitting terhadap old habit yang mungkin sedikit banyak kita longgarkan pada waktu zaman sebelum pandemi. Kemudian juga kita juga melakukan pembatasan sosial, kita sebar shift pekerjaannya di pabrik-pabrik mungkin yang awalnya shift-nya 8 jam, kemudian dikasih jeda 1 jam antar shift, atau mungkin juga bisa diberikan penyebaran jadwal produksi yang awalnya mungkin Sabtu Minggu libur, karena untuk mengurangi jumlah orang yang bekerja dalam satu waktu, kita akan sebar jadwalnya. Dan yang kemudian juga nggak kalah pentingnya, kita lakukan segregasi terhadap area-area yang memang menjadi titik kumpul. Contohnya kafetaria ataupun kantin, kemudian changing room, dan hal-hal lain yang memang nantinya bisa membantu kita untuk mengurangi potensi penyebaran virus COVID-19 tersebut. Ini adalah pendekatan kita terhadap personal protection. Kemudian yang kedua juga kita lakukan contactless operation. Absennya mungkin dulu masih pakai fingerprint. Ke depan sekarang banyak yang ketika absen kita menggunakan RFID ataupun menggunakan face recognition. Kemudian juga terkait dengan soap dispenser ataupun dispenser sanitizer, itu kita pakainya otomatis, jadi tidak perlu pencet, sehingga mengurangi potensi kontak meninggalkan virus di satu alat ataupun suatu equipment. Dan yang ketiga terkait dengan people tracking dan juga tracing. Kita juga diminta untuk memastikan kondisi kesehatan kita setiap hari sebelum kita masuk ke kantor, terus kemudian juga di kantor ataupun pada setiap hari itu ketemu orang siapa saja, sehingga pada saat nanti kita ditemukan ada potensi terkontaminasi virus COVID ataupun juga melakukan isolasi, itu kita bisa langsung men-trace. Orang ini kemudian pada waktu itu ketemu kontak erat dengan siapa aja nih. Jadi kemudian langsung bisa kita isolasi orang-orang tersebut. Diharapkan data itu sudah ada. Jadi untuk tracing dan juga tracking bisa membantu untuk melakukan self-isolation ataupun isolasi yang bisa dilakukan secara cepat. Dan juga pada waktu kemarin ataupun sekarang juga masa pandemi, kita juga melarang berbagai macam kunjungan, terus kemudian juga kalau yang memang tidak signifikan, tidak esensial, itu ditangguhkan dulu kunjungan-kunjungan ke area industri. Ini adalah terkait dengan people ataupun personal protection. Nah, hal yang kedua yang juga harus kita siapkan ataupun harus kita adaptability untuk adapt untuk keadaan yang kemarin pada saat pandemi, ini adalah mengenai supply chain disruption. Lockdown terjadi di banyak negara. Ketika negara-negara seperti China, India melakukan lockdown, kita sangat kelimpungan mendapatkan bahan baku karena majority bahan baku kita adalah berasal dari dua negara tersebut. Kemudian juga kita juga rasakan yang namanya distribusi juga terhambat. Ada flight restriction, ada container shortage, ada ketika kita barangnya udah dapat tapi kita nggak dapat vesselnya, nggak boleh berpindah dari negara satu ke negara lain, dan juga ada minimum workforce di ports dan juga kargo. Ini benar-benar sangat challenging untuk menjaga kelangsungan bisnis kita. Nah, industri pada saat kejadian seperti itu harus agile terhadap keadaan-keadaan supply chain disruption tersebut. Apa saja yang bisa kita lakukan? Kita sebenarnya bisa mulai dengan melakukan risk assessment terhadap semua rantai pasokan kita. Mulai dari hulu ke hilir, kira-kira material apa saja yang kita punya resiko untuk bermasalah ataupun shortage, terus kemudian juga distribusinya material tersebut seperti apa, kemudian secara stoknya kita inventorinya bagaimana, kemudian juga distribusi produk kita sampai ke tangan end customer. Kita lakukan risk assessment semuanya dan juga mitigasi resiko dari semua poin rantai supply chain tersebut. Nomor dua, diversifikasi juga memainkan peranan penting, di mana pada saat satu negara di lockdown, sebisa mungkin memang kita punya second source, yang ini membuat strategi ketika kita menentukan second ataupun third source, itu sebisa mungkin memang berbeda negara ataupun berbeda area, sehingga ketika suatu daerah ataupun suatu negara terkena impact lockdown, kita masih punya supply dari area yang lain. Nah, ini juga penting untuk menjadi strategi kita bagaimana melakukan diversifikasi selain dari sisi bisnis. Nomor 4, kita menghitung ulang nih recalculation terhadap buffer stock dan juga DOI. Pada saat pandemi keadaan tidak menentu, mungkin yang paling ideal memang awalnya kita zero inventory. Tapi kemudian ketika masa pandemi ini kita rebutan, bukan berebut ya, maksudnya kita saling berlomba untuk mendapatkan bahan baku, terus kemudian juga tidak ada kepastian supply. Pada saat ada kesempatan dan juga supply itu ada, kita harus menghitung ulang apakah kita perlu menaikkan buffer stock kita dibandingkan dengan NOC kita atau net operating cycle kita, operating cash kita, uang ataupun modal yang kita harus keluarkan karena kita menimbun stok dalam tanda kutip. Juga dibandingkan juga dengan business opportunity yang mungkin bisa kita sasar ketika kita punya stok tersebut. Ini keuntungannya sebenarnya apa sih? Nah, ini semua adalah strategi-strategi yang bisa kita lakukan untuk memastikan bisnis kita tetap berjalan walaupun secara supply chain mengalami hambatan. Dan yang terakhir tentu flexible manufacturing schedule, di mana memang pada saat kemarin terasa banget, sampai sekarang pun juga masih sedikit terasa terkait dengan kita flexible manufacturing schedule, mana yang memang barangnya ready, mana yang barangnya dibutuhkan, itu yang kita perlukan dari sisi bisnis, dan kita bisa agile terhadap perubahan-perubahan seperti itu. Yang nomor ketiga, terkait dengan adaptability, yaitu mengenai peluang. Jadi di satu sisi yang tadi disampaikan mengenai COVID-19 kita kelihatannya banyak challenges, ternyata di sisi yang lain menimbulkan peluang nih, yaitu mengenai new market opportunity. Bagaimana pengembangan vaksin di seluruh dunia ini bisa berpacu semuanya untuk menghasilkan vaksin dan juga menjual vaksin mereka ke seluruh dunia, karena baru kali ini mungkin seluruh dunia membutuhkan hal yang sama, makanya mereka berlomba-lomba untuk menghasilkan vaksin. Kemudian juga kita alami rediscovering new indication, obat-obat yang dulunya common, kemudian ternyata juga bisa dipakai untuk meringankan gejala-gejala COVID-19. Dan di tahun kemarin juga kita rasakan bagaimana percepatan registrasi obat yang dilakukan oleh regulatori. Produk-produk yang awalnya mungkin untuk pendaftaran bisa sampai 1 tahun, ataupun paling cepat 7 bulan, 8 bulan, ini kemarin bisa sampai 2 bulan keluar registration number-nya. Nah, ini pun juga opportunity ini yang harus kita lihat sebagai sebuah kesatuan bisnis untuk tetap mempertahankan ataupun menjaga keberlangsungan bisnis kita. Dan yang terakhir mirip dengan yang tadi, yaitu mungkin kita karena chronic disease kebutuhannya memang tidak sebesar mengenai COVID-19 yang related drugs-nya, kita shifting. Pada momen-momen tersebut kita harus agile, shifting mana yang memang lebih dibutuhkan, sehingga bisnis kita tetap berlanjut. Nah, itu semua adaptability, reaktif kita terhadap keadaan semasa pandemi kemarin, semasa COVID-19. Nah, kalau ke depan terjadi lagi, apa sih yang sebenarnya, apakah kita sebenarnya siap nggak kalau misalnya ke depan terjadi ketidakpastian seperti pandemi seperti sekarang ini? Jawabannya adalah sebenarnya para ahli di luar sana sudah mengindikasi ataupun melakukan memulai sebuah studi terkait dengan yang disebut Pharma 4.0 dan juga Quality 4.0. Ini untuk mendukung percepatan menuju industri 4.0 yang sudah sering kita dengar selama ini. Padahal kita tahu bahwa industri farmasi ini adalah industri yang highly regulated. Kita bukan industri otomobil yang ketika perubahan itu terjadi bisa dilakukan secara big bang, langsung cepat dan juga besar. Tapi kita karena kita highly regulated, kita melalui berbagai macam tahapan dan juga melalui berbagai macam kajian risiko, sehingga industri 4.0 ataupun yang pharma dan juga quality 4.0 ini bisa juga diterapkan di dalam industri farmasi. Di tahun 2017 dari studi yang dilakukan oleh FDA diketahui bahwa quality issue menyumbang peran terbesar terhadap kelangkaan obat. Quality issue ini bisa terjadi karena memang limit pengetahuan kita terhadap penyebab dan juga variasi ataupun apapun itu belum clear, sehingga keterbatasan ataupun penyediaan obat ini menjadi terhambat. Ini juga yang kemudian dirasakan pada saat sekarang, yaitu sekarang ini kita menganut sistem adalah current single submission based control strategy, di mana critical quality attribute-nya terpenuhi, quality target product profile-nya terpenuhi, itu sementara sudah cukup. Tapi yang belum dipertimbangkan adalah yang dari sisi sebelah kanan. Apa sih sebenarnya perubahan-perubahan yang terkait dengan GMP, facility, utility yang menyebabkan variasi dalam produknya? Apakah kemudian pengaruh dari material, critical attribute, impurity, dan lain-lainnya terhadap complete life cycle produknya? Kemudian juga kontrol strateginya bagaimana? Data integrity-nya pada saat pengembangan sampai dengan pendaftaran itu dan juga life cycle produknya itu seperti apa. Semua hal ini sebenarnya belum dipertimbangkan menjadi salah satu poin yang menjadi pertimbangannya si regulatori. Padahal kalau kita lihat di FDA, di situ untuk pharmaceutical industry di abad 21 diharapkan pharmaceutical industry ini adalah agile, efisien, dan fleksibel yang bisa menghasilkan produk yang berkualitas dengan kata kuncinya adalah tanpa extensive regulatory oversight. Maksudnya adalah tanpa kontrol dari regulatori yang sangat berlebihan. Jadi diharapkan memang semua industri ini mandiri dalam pengembangan life cycle produknya, dari mulai pengembangan sampai produk discontinuation, agar kualitinya ini bisa tercapai. Dan ini pun juga di ICS juga sudah ditentukan ataupun sudah dibantu dengan klausul-klausul dari Q8 sampai Q14 terkait dengan kontrol terhadap keseluruhan life cycle produknya. Kalau kita kemudian ditanya seperti apa sih Pharma 4.0, apakah itu sebuah produk yang kita bisa beli di pasar atau sebuah sistem yang kita bisa download di internet? Nah, Pharma 4.0 itu lebih kompleks dibanding itu. Pharma 4.0 ini merupakan tahapan proses yang kita dari awal step pertama kita siapkan terkait dengan people-nya, physical system-nya, data, untuk memastikan ataupun meningkatkan dari sisi quality, productivity, dan juga profit-nya. Kemudian di step kedua, kita fokus terhadap 3 prinsipal elemen, yaitu adalah terkait dengan intelligence, dan juga connectivity, dan juga flexible automation. Sehingga harapannya, Pharma 4.0 ini bisa menjadi sebuah sistem yang memprediksi. Jadi bukan lagi korektif, bukan lagi preventif, tapi dia memprediksi ketika ada masalah. Dan dia bisa memberikan solusi yang autonomous, yang dia bisa memberikan autonomous solution di step 4, yang sejalan dengan human centric, sehingga berhubungan dengan creativity, problem solving, dan juga entrepreneurial spirit. Ini adalah rangkaian proses yang harus kita lalui untuk mencapai Pharma 4.0. Jadi bukan seperti kita beli mesin, plug and play, kemudian selesai, bukan seperti kita download sistem, kemudian kita implement, selesai, enggak. Tapi stepnya banyak, dari awal sampai akhir, mulai dari penyiapan people, physical system, dan juga datanya, kemudian juga fokus ke 3 main principle, intelligent, connectivity, dan flexible, sehingga harapannya sistem yang kita bangun ini, ekosistem yang kita bangun ini, bisa membuat ekosistem yang prediktif dan juga autonomous. Jadi fully autonomous. Ini harapannya menuju Pharma 4.0, kalau di masa lalu regulator menerimanya adalah annual product quality review. Di masa yang depan, semakin kita mendapatkan banyak data, semakin kita mengetahui semua faktor dan juga elemen dari product life cycle dengan continuous process verification ataupun continuous product monitoring dengan real time monitoring, dan digabungkan dengan MES, ERP, PLM, dan juga training. Ini akan meningkatkan tekanan ke tim R&D untuk pengembangan produknya nggak cuma cepat, tapi juga mengarah ke personalized therapy. Nah, ini adalah Pharma 4.0 di masa depan. Jadi harapannya memang tujuannya sampai ke arah situ. Ini kata kuncinya, old ways won't open new doors. Jadi kalau kita menggunakan cara yang lama, tentu kita akan menghasilkan hasil yang sama. Makanya kita harus mulai untuk sesuatu yang berbeda dan juga sesuatu yang baru. Di ISPI, saya menyadul dari ISPI terkait dengan Pharma 4.0 ini ada 2 enabler dan juga 4 elemen. Yaitu enablernya adalah data integrity by design dan juga digital maturity. Sedangkan elemennya adalah kita siapkan secara resources-nya, digitalisasi dan workforce 4.0-nya available dan juga qualified. Information system-nya holistic, value network, integration, dan juga bisa di-trace. Culture-nya mengenai communication, decision making-nya juga pasti akan berbeda. Dan juga yang paling penting adalah terkait dengan holistic control strategy life cycle management. Ini adalah perpaduan dengan ICH di Q10 di Pharmaceutical Quality System. Jadi kalau disamarikan, seperti apa sih Pharma 4.0 ini? Ini dibangun dari semua mesin, semua sensor, dan juga komputer, peralatan, dan lain-lainnya, yang nantinya si mesin ini bisa melakukan prediktif, bisa melakukan self-aware component, atau dia tahu nih kapan sensor tersebut akan rusak, kapan mesin tersebut akan breakdown, kapan akan ada penyimpangan, dan lain-lainnya. Dan yang nggak kalah pentingnya ketika kita masuk ke Pharma 4.0 adalah mengenai big data. Yang sekarang kita mungkin masih terpecah-pecah antar bagian, dari mulai produksi, di inline inspection, testing, monitoring, engineering, semuanya masih terpecah-pecah. Nah, ke depan di Pharma 4.0 ini adalah akan dikumpulkan menjadi satu kumpulan big data, dianalisa menggunakan artificial intelligence, dan juga algoritma yang akan membantu kita untuk mengeliminasi ataupun menghindari terjadinya masalah. Terkait dengan kontrol strateginya, kalau di masa lalu kita punya EPC ataupun Advanced Process Control, EPC ini lebih ke reaktif. Kalau ada masalah, kemudian kita reaktif menyelesaikan. Di Pharma 4.0, kata kuncinya yang tadi disebutkan adalah prediktif, yaitu kita melakukan monitoringnya real time, dan juga kita melakukan prediktif analisis. Kira-kira dari semua data, penurunan fungsi dan lain-lainnya, apa yang bakal menyebabkan masalah. Nah, disinilah kunci kontrol strategi kita. Di masa depan, udah bukan lagi sensor kemudian memberikan, oh ini ada masalah nih, itu adalah masih reaktif. Tapi harapannya di Pharma 4.0, ini udah lebih ke prediktif. Sebelum masalah itu muncul, dia bisa memberikan prediktif apa yang akan terjadi di masa depan. Ini saya akan skip karena sama-sama seperti yang tadi, di masa lalu hanya annual product review, di masa depan memang harapannya lebih ke continuous real-time monitoring. Nah, sehingga dengan penerapan Pharma 4.0 ini, terciptalah yang disebut Future Pharma Company, yang memegang 3 elemen penting, yaitu ini adalah responsiveness kita terhadap kemajuan zaman, terhadap industri 4.0, jadi ada new teknologi, testing, research, digitalization, dan lain-lainnya. Ini adalah respon kita untuk menjawab tantangan zaman sekarang. Terus kemudian juga kita juga menyiapkan external issue yang kemungkinan besar juga mungkin masih bisa akan terjadi di masa depan. Contohnya seperti yang kita alami sekarang, pandemi-pandemi yang mungkin akan terjadi, terus kemudian juga ada kemungkinan politik dan juga ekonomi yang berubah, yang tidak pasti, harga, market growth, dan lain-lainnya. Dan Future Pharmaceutical Company dengan menggunakan metode Pharma 4.0 ini adalah sebuah boleh dibilang opsi jawaban kita bahwa kita bukan seperti industri kebanyakan yang big bang changes itu bisa dilakukan, karena kita adalah highly regulated pharma, dan kita banyak poin-poin yang harus kita penuhi untuk bisa mencapai kemandirian ataupun autonomous system. Ini kurang lebih gambarannya mengenai industri capability di Pharma 4.0. FOF itu adalah Factory of the Future. Jadi dari sisi prosesnya bagaimana? Apakah kita melakukan shorter product changeover, kemudian continuous processes, di sisi modelnya bagaimana industri kita? Apakah modular, otomatis, robotik? Operational excellence-nya bagaimana? Ada business process, technical process, IT support, database. Good engineering practice-nya juga terkait dengan project management dan juga cost. People-nya, workforce for the people-nya, ini juga memegang peranan penting, yaitu terkait dengan teknologi, statistik data, knowledge understanding dari facility dan juga production processes. Dan tahapan prosesnya pun juga kita harapkan compliance, agile, fleksibel, dan juga meningkatkan safety dan juga productivity. Ini adalah gambaran dari Factory of the Future. Nah, dari PwC sendiri juga menerapkan model untuk mengevaluasi industri capability terkait dengan pharmaceutical company. Dari sisi production process, production quality, production cost, production cycle, dan juga production personnel. Dari sisi production quality, apakah kemudian quality validity dari quality controlnya ini sudah dijalankan? Kemudian apakah setup automatic controlling system ini sudah tersedia. Dari sisi production cost-nya, kita mengevaluasi opportunity dari material cost, optimasi dari layout plan-nya, sehingga tidak ada gerakan waste yang berlebih. Kemudian juga kita mengurangi transportation cost. Dari sisi production cycle-nya, kita mengurangi production material turnaround, ataupun kalau boleh dibilang DOI-nya. Kemudian reduce non-value added activity, ini pasti. Kemudian reduce procedure in operation process. Dari sisi production personnel, kita lakukan yang namanya streamline training plan dan juga increase training quality. Dan juga clarify roles dan responsibility. Maksudnya adalah kita memperjelas poin-poin job desk dari masing-masing role, dan juga kita pastikan secara kapabilitas dan juga kompetensi dari setiap personilnya adalah mumpuni. Dari sisi production prosesnya, kita melakukan optimasi dari production process, kita hilangkan ataupun kurangi bottleneck proses kita. Kemudian kita juga melakukan dari production life structuring dan juga optimize production process. Ini adalah model untuk mengevaluasi kapabilitas kita terhadap production management untuk kesiapan kita di penerapan Pharma 4.0. Nah, ini adalah tahapan prosesnya terkait dengan pencapaian digital maturity di Pharma 4.0. Kalau boleh disampaikan, ada dua tahapan besar, yaitu mengenai digitalisasi dan juga Pharma 4.0-nya sendiri. Kalau digitalisasi ini terdiri dari dua tahapan, yaitu adalah komputerisasi dan juga connectivity. Komputerisasi ini maksudnya adalah yang awalnya kita paper base, kita ubah rekodnya ke electronic base. Terus kemudian juga kita setiap bagian untuk data yang ada keluar dari setiap bagian ini kita hubungkan. Jadi mirip sama seperti yang dilakukan oleh ERP sebenarnya. Tapi kalau di Pharma 4.0 ini udah jauh lebih advance dibanding itu. Jadi lebih maju dibanding apa yang dilakukan oleh ERP. Yaitu tujuannya adalah di step ketiga kita bisa menerapkan yang disebut visibility. Yaitu kata kuncinya ataupun pertanyaan kuncinya adalah what is happening. Apapun yang terjadi di shop floor, apapun yang terjadi di lapangan, bisa langsung terlihat oleh manajemen. Nah, ini adalah tahapan ketiga dari Pharma 4.0. Kemudian di tahapan keempat, yaitu adalah transparansi. Kenapa itu bisa terjadi? Jadi datanya sudah ada, kemudian kita tahu bahwa itu ada masalah, analisanya pun juga bisa dilakukan. Ini adalah masih di tahapan keempat. Tahapan kelima ini jauh lebih advance dari tahapan keempat. Sebelum masalah itu terjadi, kita bisa memprediksikan, yaitu predictive capability. What will happen? Maksudnya di sini adalah sebelum masalah itu muncul, sistem yang kita bangun di Pharma 4.0, data yang muncul dari setiap tahapan proses, dari setiap mesin, dari setiap instrumen, setiap sensor yang ada, itu bisa mempredik apa yang akan terjadi, penyimpangan apa yang bakal muncul, dan juga masalah apa yang kemungkinan besar akan terjadi pada saat proses industri. Di tahapan yang paling tinggi adalah terkait dengan adaptability autonomous system, yaitu bagaimana autonomous response bisa tercapai. Ini benar-benar tahapan yang paling high, paling tinggi, di mana autonomous di sini bukan berarti kemudian kita punya mesin, kita klik mesinnya, kemudian jalan otomatis, itu masih belum otonomus. Karena ketika nanti mesinnya jam, ketika nanti mesinnya bermasalah.
Nggak bisa.
Sorry, yes, saya lanjutkan. Tadi mungkin ada yang mau ikut nimbrung ngomong, nggak apa-apa. Jadi, otonomus ini bisa kita dapatkan, maksudnya dengan kapanpun itu kemudian dia bermasalah, dengan data yang ada, dengan equipment yang ada, dia bisa melakukan tindakan perbaikan dan juga autonomous system ataupun autonomous response. Nah, dari semuanya itu, digitalisasi ini keuntungannya cukup banyak. Dari paperless production, real-time review, right first time, tighter quality control, tighter control for control quality, audit readiness, dan lainnya. Sehingga harapannya semua data ini bisa kita kumpulkan dan juga mencapai yang namanya holistic control strategi dari sisi produk, quality, dan juga cost. Dan target endingnya adalah parametric release, real-time release testing, PAT, itu semuanya bisa tercapai, termasuk juga yang nggak kalah pentingnya adalah cost effectiveness. Ini terkait dengan data integrity by design. Seperti yang kita ketahui, data integrity menjadi momok untuk semua industri, jadi sasaran empuk semua regulator ketika audit. Makanya ketika kita mau masuk ke digitalisasi, salah satu enabler yang paling penting adalah data integrity by design. Maksudnya adalah dari awal kita mendesain proses, flow proses kita harus juga mengikutkan data integrity by design kita. Bagaimana data ini akan bergerak dari satu proses ke proses yang lainnya, bagaimana data ini di-create, dan juga keseluruhan life cycle dari kehidupan si data ini, dari mulai terbentuk sampai nanti di-archive, itu menjadi sebuah hal yang harus dipikirkan juga ketika kita membangun proses. Makanya kita jangan lupakan juga ada 4 faktor penting, yaitu tentu adalah koala principles. Koala mungkin kalau teman-teman biasanya pahamnya Alcoa, cuma saya ubah saja jadi koala. Terus kemudian juga dari sisi data life cycle-nya, security dan juga safety-nya bagaimana. Dari sisi data quality-nya seperti apa kita harus juga tentukan, dan juga mengenai kebenaran datanya. Seperti yang kita ketahui ketika kita bermain dengan digitalisasi dan juga teknologi, kalau data yang kita input sampah, keluarnya akan sampah. Makanya right content ini juga harus menjadi poin penting. Ini yang tadi sepintas saya sampaikan, jadi data ini akan berpindah dari semua proses, kemudian untuk menerapkan data integrity ini tentunya pasti akan sebuah challenge yang sangat besar, baik ini yang masih sekarang paper based ataupun besok yang udahan digital based. Kemudian juga asal-usul datanya juga harus lebih transparan, dan yang paling penting tadi di environment yang kita bangun, flow proses mulai dari proses awal sampai proses akhir, jangan lupa untuk juga menerapkan ataupun menginput apa sih yang harus kita lakukan untuk menjamin data integrity di setiap prosesnya. Nah, terus kemudian juga terkait dengan holistic control strateginya, red thread atau benang merahnya tuh harus nyambung antara mulai dari design, implementation, dan juga operation of control strategi. Nah, ini semuanya adalah cakupan yang terkait dengan data integrity by design. Sehingga harapannya dengan tadi data integrity by design kita sudah capai, terus kemudian dari sisi capability produksinya ataupun industrinya juga sudah kita penuhi, terus kemudian dari sisi digital maturity-nya udah pelan-pelan kita capai. Harapannya memang Pharma 4.0 ini udah bukan mimpi lagi. Makanya harus mulai dari sekarang kita mulai untuk masuk ke teknologi ataupun masuk ke tatanan dunia yang baru. Yang itu memang sudah juga digaungkan oleh seluruh industri farmasi di dunia. Nah, kalau di Quality 4.0 ini adalah tindak lanjut ataupun penyempurnaan dari ICH Q10 dan terkait dengan pharmaceutical quality system yang di sini juga ditambahkan ataupun diperkaya dengan elemen dan juga enabler sama seperti yang tadi disampaikan di Pharma 4.0. Jadi kalau yang dulu di PQS yang lama ataupun di current PQS, kita mengenal proses performance dan juga product quality management system, kappa system, change management, management review. Kemudian itu akan berjalan di seluruh life cycle produknya, mulai dari development, transfer tech, komersial, sampai product discontinuation. Ini ditambah dengan new PQS elemen di Pharma 4.0, yaitu terkait dengan data integrity, process maps, integrated planning, environmental monitoring dan energy management, automation, termasuk di dalamnya PAT, CPV, CPM, RTRT, dan juga parametric release, dan didukung juga dengan knowledge management dan juga quality risk management. Sehingga poin-poin pentingnya terkait dengan product dan proses understanding, QBD, dan juga interdisciplinary collaboration, juga end-to-end integration dari semua GXP, ini akan terjadi. Dan Nanti juga jangan lupa dari sisi quality system integrated-nya, MIS, PLM, semuanya ini menjadi satu, sehingga tercapai yang disebut dengan life cycle management untuk holistic manufacturing control. Ini poin-poin kenapa sih kita perlu untuk menerapkan Quality 4.0 ataupun apa sih keuntungan yang bisa kita dapatkan dari penerapan Pharma 4.0, Quality 4.0, sorry. Yaitu ada 5 sebenarnya. Dari nomor satunya adalah increase scalability, maksudnya adalah dengan kita menerapkan Quality 4.0, ini setiap kali ada perubahan, kita mungkin nggak harus melakukan revalidasi untuk keseluruhan area ataupun keseluruhan sistem, tapi kita cuma memvalidasi apa yang berubah. Ini akan sangat-sangat membantu dari sisi flexibility industri kita. Kemudian juga greater visibility of risk across product life cycle. Ini maksudnya adalah apapun yang terjadi di upstream pada saat pengembangan produknya, ini semua bisa kita antisipasi sehingga tidak menjadi masalah dari sisi downstreamnya pada saat produk commercial manufacturing. Ini kata kuncinya. Increase agility on the shop floor, tentunya kita juga menerapkan ataupun meningkatkan terkait dengan kompetensi shop floor-nya, bagaimana mereka yang awalnya paper based menjadi digital based, dan connecting the manufacturing ecosystem ini lebih ke horizontal connecting antar bagian. Jadi semua yang terjadi di satu bagian ini kita akan tahu dampaknya ke bagian yang lain dan juga apa yang harus dilakukan oleh bagian yang lain, sehingga end-to-end prosesnya ini benar-benar bisa tercapture, dan apa ataupun semua tindakan yang kita lakukan benar-benar bisa membantu untuk mencapai ataupun menyelesaikan masalah dengan lebih cepat. Nah, yang terakhir terkait dengan kolaborasi dengan global partner dan juga supplier. Seperti yang kita ketahui, kadang kita juga kesulitan untuk mengontrol quality dari sisi global partner dan juga supplier. Tapi dengan penerapan Quality 4.0 ini, harapannya memang kita bisa lebih mengontrol terkait dengan kapasitas sistem ke merekanya, terus kemudian bagaimana mereka audit readiness-nya di global partner kita. Ini sangat membantu kalau kita memang akan menerapkan Quality 4.0. Nah, new basic konsepnya dari quality assurance ada 2, yaitu adalah adaptasi terkait sama cross-functional bisnis prosesnya, dan juga kita mempercepat decision making terhadap suatu problem. Ini tentu adalah 2 hal yang memang menjadi keuntungan dengan kita menerapkan Quality 4.0. Untuk terkait dengan workforce capability atau dari sisi HR-nya, kompetensi dari sisi teknologi, statistik, industrial knowledge, software management, change management, dan juga new culture dan juga behavior, ini semuanya menjadi penting untuk dipunyai oleh workforce, the people-nya, ataupun secara human resources-nya. Ini yang juga harus kita siapkan. Sehingga nanti akhirnya akan tercapai ekosistem pharma dan juga Quality 4.0 dari semua departemen, dan juga big data-nya terkait dengan compliance, lead time, cost control, safety, semuanya akan menjadi satu dari semua berbagai macam jenis proses yang ada di setiap bagian. Jadi memang kompleks pharma dan juga Quality 4.0 ini sebuah sistem, sebuah ekosistem yang nantinya akan pelan-pelan harus kita semua mengarah ke sana. Sehingga dengan penerapan Pharma dan juga Quality 4.0, kita bisa siap kalau ada ketidakpastian di masa mendatang. Konklusinya adalah coronavirus ini akan menjadi, efeknya ini akan berlangsung lama. Jadi kita harus adapt dan juga kita harus siap untuk memastikan bisnis kita tetap berjalan. Padahal kita juga di satu sisi yang lain, kita juga harus menyiapkan safety terhadap para pekerja kita, dan juga terkait dengan adaptation new normal, kita akan menyiapkan ataupun meningkatkan opportunity untuk menerapkan dan juga mensupport industri 4.0, dan dengan adopting new digital technology kita akan mempercepat proses, improve flexibility, creativity, dan juga compliance. Ini semua adalah konklusi dari apa yang saya coba sampaikan pada hari ini, dan Ini adalah referensinya. Sebelum saya tutup, sebenarnya saya ada satu video. Semoga ini bukan iklan videonya. Jadi kita akan coba tampilkan videonya. Bentar ya. Semoga ini saya lihat pada waktu itu adalah iklan mengenai drone kamera DJI. Mungkin teman-teman sudah paham ya. Jadi bentar. Saya siapkan ininya dulu suaranya. Share komputer sekarang. Nah,
For as long as
kita
people have
lihat
walked the
ini mungkin
earth,
teknologi di masa
they've
depan.
wondered how to fly.
Buat teman-teman
spent
yang
more than a
lihat
decade
video
crafting
ini, ketika
tools
saya
for
lihat, oh mungkin
dreamers.
teknologi masa depan akan
Now,
seperti
we're
ini.
going one
Orang yang
step
takut terbang,
beyond
mungkin
with
sebenarnya
an
secara
immersive
fisik nggak perlu terbang.
experience unlike anything else.
Dengan bantuan
Introducing
drone seperti
DJI
ini, kita bisa langsung
FPV.
merasakan efeknya ketika kita terbang. Jadi menggunakan Google, bagaimana mereka akan
The world's first
bisa
ready-to-fly
merasakan hal tersebut.
FPV drone that lets users of
Cuma
any
contoh
skill
kecil dari
level feel
bagaimana
the
industri
thrill
ini
of
benar-benar
immersive
atau
flight.
teknologi ini benar-benar mengubah tatanan kehidupan. Mungkin suatu saat nanti di masa depan
We've
kita nggak
also
perlu pergi ke luar
revolutionized
negeri,
the
cuma
FPV
baik dengan kacamata
experience
seperti ini kita
by
benar-benar
creating
sudah
the
bisa juga
DJI motion
merasakan
controller,
hidup di luar
a
negeri itu seperti
compact
apa sih.
device that enables ultra-intuitive
Dan
flight
mimpi kita
based
mungkin
purely on hand motions. With
kembali
the
ke masa
standard
lalu
remote
ataupun
controller,
apapun itu,
3 distinct flight modes
kita juga
offer
bisa
multiple
seperti itu.
options for
Jadi
anyone to fly
ini adalah
FPV.
benar-benar yang
Start
dirasakan
with N mode for an
oleh
immersive experience with
ataupun yang
traditional flight
di
controls.
luar itu ternyata teknologi sudah
S mode
dipakai
simplifies
seperti
FPV controls for an exciting
kita
hybrid
benar-benar merasakan
flight.
seperti terbang. Jadi mimpi manusia terbang mungkin nggak jadi mimpi lagi dengan cara seperti itu.
Activate M mode to unlock unlimited flight
Saya
controls.
cukup ya. Saya
Now
bukan
the only
lagi
boundaries are your
jadi
skills
akan
and
tutup
imagination.
videonya. Saya kembali ke slide presentasinya. Demikian penutup dari saya. Jadi di masa uncertainty sekarang ini sebenarnya kita ada akses yang tak terbatas terhadap teknologi yang bisa membantu karyawan, perusahaan, community, dan juga pemerintah untuk terus maju ke depan. Demikian presentasi dari saya. Terima kasih atas perhatiannya.
Oke, terima kasih banyak Bapak Surya atas presentasinya dan juga sharing knowledge-nya untuk kita semua. Ada pertanyaan dari Google Form, anonimus untuknya pertanyaannya, jadi saya tidak bisa memberitahukan ini siapa yang menanyakan. Jadi pada masa pandemi, kan terdapat pembatasan jumlah orang, bagaimanakah solusi dari pihak industri ketika ingin melakukan hal seperti misalnya uji klinis yang mungkin perlu mengumpulkan jumlah orang? Bagaimanakah mekanismenya dari uji klinis dari obat yang akan dilakukan pada masa pandemi? Begitu juga dengan pemilihan subjek, ujinya dilakukan seperti apa, dan apakah ada kriteria khusus yang berbeda dari uji klinis sebelumnya? Terima kasih.
Oke, saya coba jawab. Jadi memang dari survei yang dilakukan oleh PwC juga sudah disampaikan bahwa COVID-19 ini benar-benar mengganggu rekrutmen for clinical research dan juga akan sangat mengganggu terkait dengan launching produk. Memang saya pun juga mengalami terkait dengan hambatan melakukan clinical research. Jadi kalau di masa pandemi seperti sekarang ini, clinical research ini bukan menjadi yang prioritas. Makanya karena keadaannya emergency pandemi, memang kita lebih adaptif terhadap kondisi seperti itu. Kemarin berdasarkan pengalaman, ada berbagai macam pengalaman, yaitu ketika melakukan clinical research ini, kemudian kita bisa melakukannya secara separasi ataupun secara segregasi sebenarnya, dan itu masih memungkinkan sebenarnya. Jadi pengambilan datanya dan juga semua kondisinya itu dilakukan dengan risk management dan juga secara terkontrol, sehingga kalau memang ini clinical research-nya untuk mendukung COVID-19, saya yakin pasti akan dikebut. Tapi kalau memang bukan untuk COVID-19, memang challenging untuk bisa di ACC terkait dengan sebuah kondisi yang nggak normal tadi seperti tadi. Kalau terkait dengan kondisi apakah pandemi ini menjadi pembeda terkait dengan kondisi orang yang akan menjadi subjek clinical research, tentu untuk produk-produk tertentu ya, karena COVID-19 ini menjadi sebuah pandemi, dia bisa menjadikan apa ya sebutannya, sebuah studi baru terkait dengan keberhasilan suatu obat. Dan juga untuk pengembangan produk sebenarnya dari sisi EU itu sudah menyetujui penggunaan artificial intelligence sebagai gantinya pelaksanaan desain space yang untuk yang biasa dan juga untuk waktu yang lama. Ya, untuk biayanya jadinya juga lebih ditekan. Jadi memang sekarang arahnya ke arah sana. Memang artificial intelligence juga memainkan peranan penting. Kalau kita lihat di EU sudah arahnya juga menuju ke arah sana. Tapi kalau kita bicara di Indonesia, memang masih challenging. Itu.
Oke, baik. Terima kasih banyak, Pak Surya. Mungkin karena waktunya sudah singkat, saya akan segera akhiri sesi ketiga daripada acara PDA e-Townhall ini. Sekali lagi kami dari I3L dan juga dari PDA dan juga dari pihak panitia mengucapkan terima kasih sebanyak-banyaknya atas kesediaan Pak Surya untuk membagi ilmunya dan juga membagi pengalamannya, terutama dalam topik bagaimana mengadaptasi pandemi pada industri farmasi tentunya. Kepada para peserta, sebelum kita mengakhiri sesi ini dan juga mengakhiri event ini, jangan lupa untuk mengisi evaluation survey, karena nanti sertifikatnya hanya akan diberikan kepada pihak yang mengisi evaluation survey tersebut. Kemudian sertifikat dapat didapatkan kira-kira antara 7 sampai 15 hari kerja setelah webinar ini berlangsung. Sekali lagi kami dari I3L dan dari panitia mengucapkan banyak terima kasih kepada pembicara, kepada partisipan, dan kepada peserta semuanya. Kami harap kami dapat bertemu lagi dengan Anda semua di event-event kami dan juga acara kami berikutnya. Terima kasih semuanya.
Thank you, thank you.
Thank you semuanya, terima kasih banyak.